There is a system inside you that most people have never thought about, never learned the name of, and never had occasion to notice, and yet it has been working on your behalf every single day of your life, quietly, without announcement, without asking for credit, doing some of the most important and most intricate biological work the body performs. It drains the fluid that leaks from your blood vessels. It absorbs the fat from your last meal. It patrols the highways of your immune system, carrying the surveillance cells that watch for anything that should not be in the body and mounting the response when they find it. It is the lymphatic system. And it is, in almost every sense that matters, the body's unsung river. The reason most people have not heard of the lymphatic system, or have heard of it only vaguely, as something associated with swollen glands or cancer staging or the kind of massage that claims to detoxify, is that it operates below the level of conscious sensation. The cardiovascular system announces itself with every heartbeat. The nervous system announces itself with every thought and every touch. The lymphatic system makes no such announcements. It has no pump that you can feel. Its vessels are invisible through the skin. It does its work in the tissue spaces between the organs, in the lymph nodes that are only noticed when they swell, in the spleen and the thymus and the tonsils that most people can name but few could describe in any detail. The lymphatic system is the body's quiet infrastructure, and tonight we are going to slow down enough to appreciate it properly. I find that there is a particular quality of wonder available in learning about something the body does without your knowledge. A kind of retroactive gratitude, perhaps, for the work that has been done on your behalf without your ever being asked to authorize it. Every hour of every day of your life, the lymphatic system has been returning fluid to your blood, helping your immune cells travel to where they are needed, absorbing the fats that feed you and the vitamins that sustain you. And it has done all of this without a single moment of your attention. What would it mean, I wonder, to give it some attention now? In the dark, in the quiet, at the end of a day that has asked much of the body? To simply hear what it does, and to feel something like recognition? This is Relax with Science. And tonight we are going to travel somewhere remarkable together. Slowly, gently, with nowhere else to be. Into the capillaries and the vessels and the nodes and the organs of the lymphatic system. Into the biology of fluid balance and immune surveillance and the extraordinary cellular life of the lymph node. Let the room be quiet. Let the breath slow. And let the body rest in the knowledge that even now, as the day releases its last hold, the lymphatic system is doing what it has always done, quietly, faithfully, keeping the balance. I wonder where you are tonight. What kind of day the body has carried you through to arrive here? Whether the day asked a great deal of you or was gentler than most. Whether the room is quiet or whether there is still sound in the world beyond it. If you feel like it, and only if you feel like it, tell me in the comments where in the world you are right now. Just a city, or a country, or a time on the clock. I read every one. It genuinely matters to me that you are there. If these words bring you any ease tonight, any settling of what the day left in the body, a quiet like on the video is a small and generous way to say so, and it helps relax with science find its way to someone else who might need exactly this kind of company in the later hours. If you know someone who carries tiredness to bed, consider sending this to them. No explanation needed. Just the link, left where they will find it. And if there is ever something that could be softer, slower, or more beautiful about what we make here, I want to hear it. This channel belongs to the person lying in the quiet. It has always been for you. The lymphatic system is the body's secondary circulatory system, a network of vessels, nodes, and organs that runs alongside and complements the cardiovascular system, returning the fluid that continuously leaks from blood capillaries back to the bloodstream and serving as the highway of the immune system's cellular traffic. The name lymph comes from the Latin lympha, meaning clear water or water goddess, and the clarity of the liquid is one of the things that surprises people when they first encounter the lymphatic system as something more than a name. Lymph, the fluid that flows through the lymphatic vessels, is pale and watery, almost colorless in most of the body, becoming the milky chyle of the intestinal lacteals when fat-laden. It is essentially blood plasma from which the red blood cells and most proteins have been filtered out by the capillary walls, enriched with immune cells gathered from the tissues, containing the cellular debris and metabolic waste products that the blood capillaries filtration has deposited in the interstitial spaces. The lymphatic system exists because the cardiovascular system leaks. Every minute, the heart pushes blood through the capillary beds at a pressure sufficient to drive a net filtration of fluid from the blood into the surrounding tissue spaces. Approximately 20 liters of fluid per day in an average adult. Of this, approximately 17 liters is reabsorbed back into the capillaries by osmotic forces. The remaining 3 liters, carrying proteins too large to be reabsorbed by the capillaries, fat molecules absorbed from the gut, and immune cells returning from their tissue patrols, enters the lymphatic capillaries and begins the journey back to the venous blood through the lymphatic vessels, the lymph nodes, and ultimately the thoracic duct. Without this return, the tissues would progressively swell with accumulated fluid, the lymphedema that provides one of the most vivid demonstrations of what the lymphatic system does when it fails. The lymphatic system is the body's quiet corrective, the system that manages what the cardiovascular system necessarily leaves behind. The lymphatic capillaries, the smallest and most numerous components of the lymphatic vascular network, are blind-ended, thin-walled tubes that begin in the interstitial spaces of almost every tissue in the body and open to collect the fluid, proteins, lipids, and cells that accumulate there, entering the lymphatic circulation through a uniquely designed gateway structure. A lymphatic capillary, seen under a microscope, looks in some ways like a blood capillary, a tube formed by a single layer of endothelial cells. But in the details of its structure, It is radically different in ways that reflect its different function. Blood capillary endothelial cells are joined by continuous tight junctions that restrict the passage of molecules between cells to small, water-soluble substances. Lymphatic capillary endothelial cells are joined by what are called button-like junctions, discontinuous attachments at intervals around the cell perimeter that create flap-like openings between adjacent cells. These flaps function as one-way valves when the interstitial pressure rises. When fluid accumulates in the tissue, the pressure pushes the flaps inward, opening the junctions and allowing fluid, proteins, and cells to enter the lymphatic capillary. When the pressure in the lymphatic lumen is greater than in the interstitium, the flaps are pushed shut, preventing backflow. The lymphatic capillary is thus a passive, pressure-responsive collector that opens precisely when its function is most needed and closes to prevent what it has collected from escaping. The lymphatic capillary endothelial cells are also anchored to the surrounding extracellular matrix by fine filaments of collagen and fibrillin, the anchoring filaments. These filaments hold the lymphatic capillary open when the surrounding tissue swells, as edema develops and the extracellular matrix expands. The anchoring filaments pull the lymphatic capillary walls apart, maintaining the lumen open and the collecting function active even when the surrounding tissue is under physical stress. The lymphatic capillary is not simply a drain. It is an actively maintained, self-regulating drain that operates best under precisely the conditions that create the most need for drainage. The lymph nodes, the small, bean-shaped organs distributed along the lymphatic vessels throughout the body, are the immune system's filtering and surveillance stations, where lymph from the periphery is passed through a dense meshwork of immune cells that sample its contents for the presence of foreign antigens, pathogens, and cellular abnormalities. There are approximately 600 to 800 lymph nodes in the human body, distributed in regional clusters that drain specific anatomical territories. The cervical nodes drain the head and neck. The axillary nodes drain the arm, the breast, and the lateral chest wall. The inguinal nodes drain the leg, the perineum, and the lower abdomen. The mesenteric nodes, the largest cluster, several hundred strong, drain the gastrointestinal tract and are embedded in the mesentery of the small intestine. Each node is a compact, encapsulated structure, typically 5 to 15 millimeters in length, divided internally into cortex and medulla by a fibrous stroma. The lymph enters the node through multiple afferent lymphatic vessels that pierce the capsule at various points and flows through the subcapsular sinus, a space just beneath the capsule, before percolating through the node's internal architecture. In the cortex, primary lymphoid follicles, densely packed spheres of B lymphocytes, await antigenic stimulation. When B cells encounter their specific antigen, brought to the node by the lymph flowing through, the follicle transforms into a germinal center, an extraordinarily active cellular structure where B cells divide rapidly, mutate their antibody genes in the process of somatic hypermutation, compete for antigen binding, and differentiate into the high-affinity antibody-secreting plasma cells and memory B cells that constitute the humoral immune response. In the paracortex, the T cell zone between the cortex and the medulla, T lymphocytes encounter antigen presented by dendritic cells and undergo their own activation, proliferation, and differentiation. The lymph node is, in the language of immune biology, a decision-making center, the place where the immune system decides how to respond to what it has found. The thoracic duct, the largest lymphatic vessel in the body, running from the cisterna chyli in the abdomen to its drainage into the left subclavian vein at the base of the neck, collects lymph from approximately 3/4 of the body and returns it to the venous circulation, completing the circuit that allows the continuous leakage of the cardiovascular system to be perpetually corrected. The thoracic duct is the body's great lymphatic highway, a vessel approximately 30 to 40 centimeters long and 3 to 5 millimeters in diameter, far larger than any other lymphatic vessel, that gathers the lymph from the entire body below the diaphragm and the left side of the body above the diaphragm and channels it into a single convergence point at the venous circulation. It begins as the cisterna chyli, a dilated sac-like structure in the retroperitoneal space at approximately the level of the second lumbar vertebra, where the lymph from the intestinal trunk, the left and right lumbar trunks, and the intercostal trunks converges. From the cisterna chyli, the thoracic duct descends through the aortic hiatus in the diaphragm into the posterior mediastinum, running between the aorta and the azygous vein, before crossing to the left side at approximately the level of the fifth thoracic vertebra and ascending along the left border of the esophagus to the base of the neck. There it curves to the left, passes over the subclavian artery, and empties into the venous circulation at the angle of junction between the left internal jugular vein and the left subclavian vein, the left venous angle. The right lymphatic duct, shorter and more anatomically variable, sometimes consisting of a single trunk and sometimes dividing into several separate channels, drains the right side of the head and neck, the right arm, and the right thorax, emptying into the right venous angle. Together, these two ducts return the approximately 2 to 4 liters of lymph formed daily to the blood, closing the loop of fluid exchange that the cardiovascular system opens with every heartbeat. Lymph is propelled through the lymphatic vessels not by a dedicated pump. The lymphatic system has no heart, but by a combination of the intrinsic contractility of the lymphangion, the skeletal muscle pump, the respiratory pump, the arterial pulse, and the smooth muscle contractions of the larger lymphatic vessel walls. The absence of a dedicated pump is perhaps the most counterintuitive feature of the lymphatic system. How does the fluid move against gravity, from the feet to the thorax, without the equivalent of a heart? The answer is that the lymphatic system uses multiple, redundant pumping mechanisms, each contributing to the propulsion of lymph in ways that together provide reliable flow under a wide range of physiological conditions. The primary mechanism in the collecting lymphatic vessels, the vessels downstream of the capillaries, is the intrinsic contractility of the lymphangion. The lymphangion is the functional unit of the collecting lymphatic vessel, a segment of vessel between two valves, whose smooth muscle wall can contract spontaneously, propelling lymph from one segment to the next in a peristaltic sequence. The contraction is stimulated by the distension of the lymphangian wall as lymph accumulates, a stretch-sensitive response that turns the degree of filling into a contraction signal, ensuring that flow increases when formation increases. The skeletal muscle pump is particularly important in the limbs. The rhythmic contraction of the surrounding muscles during walking and exercise squeezes the deep lymphatic vessels, propelling lymph forward past the valves. This is why the lymphatic system is so dependent on physical activity. Immobility allows lymph to stagnate, and regular movement is one of the most effective physiological drivers of lymphatic flow. The respiratory pump contributes through the pressure changes of breathing. Inspiration reduces intrathoracic pressure, drawing lymph into the thoracic duct from the surrounding vessels. Arterial pulsations transmitted through the tissues provide a small additional rhythmic compression. Together, these mechanisms move approximately 2 to 4 liters of lymph per day through the body and into the venous circulation without a dedicated pump. The spleen, the largest lymphoid organ in the body, approximately 12 centimeters long and weighing 150 grams in the average adult, serves simultaneously as a blood filter, a red blood cell reservoir, and a major site of immune surveillance, with distinct red pulp and white pulp compartments carrying out these separate functions. The spleen occupies the upper left quadrant of the abdomen, tucked beneath the left side of the diaphragm and partially protected by the lower left ribs. It is a highly vascular organ, receiving approximately 5% of the cardiac output, or approximately 350 milliliters of blood per minute, and its central function is the filtration of that blood, which passes through the splenic microcirculation under conditions that make it one of the most physically demanding passages in the body for red blood cells. The spleen's internal architecture is divided into 2 functionally distinct compartments. The red pulp, which gives the cut surface of the spleen its dark red color, consists of venous sinuses surrounded by splenic cords, a loose meshwork of reticular fibers and macrophages through which the blood filters. Red blood cells must squeeze through the narrow slits between the endothelial cells lining the sinuses, slits approximately 2 to 3 micrometers wide, compared to the 8 micrometer diameter of a normal red cell, and any cell that has lost the biconcave disc flexibility required to deform and pass through these slits is trapped by the macrophages of the red pulp and destroyed. This is the spleen's quality control for red cells, aging cells, damaged cells, and cells from hemolytic conditions are selectively removed here. The white pulp, consisting of the periarteriolar lymphoid sheaths and the lymphoid follicles surrounding the central arteries, is the spleen's immune surveillance compartment, organized similarly to a lymph node but exposed directly to the blood rather than to lymph, enabling the immune monitoring of the blood's contents for circulating antigens and pathogens. The thymus, the bilobed lymphoid organ situated in the anterior superior mediastinum, above and in front of the heart, is the site of T lymphocyte maturation, where the precursor cells exported from the bone marrow undergo the education that distinguishes self from non-self and that produces the mature, self-tolerant T cells of the adaptive immune system. The thymus is one of the great developmental organs of the immune system, the school in which T lymphocytes learn what they need to know before they are released into the body. It achieves peak development in infancy and early childhood, when the demands for T cell production are highest, and begins its progressive involution, the replacement of lymphoid tissue by fat, after puberty, driven by the rising sex steroids of adolescence. The thymus receives precursor cells, thymocytes, from the bone marrow, and these cells must navigate a rigorous educational process before they are qualified for release. They begin in the outer cortex of the thymus, where they undergo positive selection, the thymocytes that express T cell receptors capable of recognizing self-MHC molecules, the cell surface proteins that present antigen to T cells, are selected for survival, while those whose T cell receptors cannot recognize self-MHC are allowed to die. Then the positively selected thymocytes migrate to the inner medulla, where they undergo negative selection, those whose T cell receptors bind too strongly to self-peptides presented on self-MHC, meaning they would react against the body's own tissues, are induced to die by apoptosis. Only the cells that emerge from this dual selection, capable of recognizing MHC-presented antigens but not reactive against self, are released as mature, naive T lymphocytes. This remarkable process, in which the immune system is taught to distinguish self from non-self by the systematic elimination of cells that fail either test, is the basis of central tolerance, the prevention of autoimmunity at the level of T lymphocyte development. The tonsils, the paired palatine tonsils in the throat, the adenoids, pharyngeal tonsil, at the back of the nasal passage, and the lingual tonsil at the base of the tongue, form Waldeyer's ring, a ring of lymphoid tissue that guards the entrances to the digestive and respiratory tracts, providing the first immunological line of defense against inhaled and ingested pathogens. Waldeyer's ring is the body's immune gateway, the ring of sentinel lymphoid tissue that positions immune cells at the portals through which the outside world most commonly enters the body. The palatine tonsils, the most familiar members of the group, the ones visible when you open the mouth wide, are oval masses of lymphoid tissue that lie in the tonsillar fossa between the palatoglossal and palatopharyngeal arches on each side of the throat. Their surface is covered by stratified squamous epithelium that dips into multiple deep crypts, the tonsillar crypts, that increase the surface area available for sampling the passing air and food. The epithelium in the crypts is particularly permeable to the passage of antigens and microorganisms into the underlying lymphoid tissue, where they are sampled by the dendritic cells and macrophages that trigger the adaptive immune response. The adenoid, the pharyngeal tonsil, sits at the back of the nasal cavity and is most prominent in young children, when it is an important contributor to the immune defense of the upper respiratory tract. The lingual tonsil forms a roughened surface at the base of the tongue, sampling the contents of the oral cavity. Together, These structures form a nearly continuous ring of immune surveillance tissue at the entrance to both the digestive and respiratory systems. Any organism or antigen that enters through the nose or mouth encounters lymphoid tissue within centimeters. The tonsillar tissue is particularly important in childhood, when the first exposures to many common respiratory pathogens occur and the immunological memory that protects adults is being built. The lymphocyte, the primary cell type of the lymphatic system, comprises two functionally distinct populations: the B cells responsible for antibody production and the T cells responsible for cellular immunity, which together constitute the adaptive immune system's capability for specific, targeted, and immunologically remembered responses. The lymphocyte is, to look at, a deceptively simple cell, a large nucleus surrounded by a thin rim of cytoplasm, roughly 8 to 10 micrometers in diameter, indistinguishable between different lymphocytes under the light microscope. This structural simplicity conceals extraordinary functional complexity. The B lymphocyte, which matures in the bone marrow, the name becoming from the bursa of Fabricius where it was first characterized in birds, is the cell responsible for producing antibodies. Each B cell expresses on its surface a unique B cell receptor, a membrane-bound antibody molecule that recognizes one specific antigen with a high affinity determined by the precise amino acid sequence of the antibody's variable regions. When a B cell encounters the antigen its receptor recognizes, it is activated, proliferates, and differentiates into plasma cells that secrete large amounts of that same antibody into the blood and tissues, and into memory B cells that persist for years and can mount a rapid, enhanced response to future encounters with the same antigen. The T lymphocyte, which matures in the thymus, is responsible for cellular immunity. It expresses a T cell receptor that recognizes antigen only when it is presented on the surface of another cell by MHC molecules, a constraint called MHC restriction that ensures T cells interact with cells rather than with free antigen. The CD4 T helper cell coordinates the immune response, activating B cells, macrophages, and cytotoxic T cells through the cytokines it secretes. The CD8 cytotoxic T cell kills cells that are infected by viruses or transformed by cancer, recognizing viral or tumor antigens presented on their surface MHC molecules. Together, these two lymphocyte populations, numbering approximately 2 trillion in the body, constitute the adaptive immune system's specific, targeted, and remembered response to every antigen the body has ever encountered. The dendritic cell, the sentinel of the adaptive immune system, patrols the peripheral tissues, continuously sampling the cellular environment for foreign antigens, and when it detects them, carries the antigenic evidence to the regional lymph node where it presents the antigen to naive T cells, initiating the adaptive immune response. The dendritic cell was first described by Paul Langerhans in 1868. In the skin, where the Langerhans cells that bear his name are a specific population of dendritic cells, and was later characterized as a distinct cell type by Ralph Steinman in the early 1970s, work for which Steinman received the Nobel Prize in 2011. The dendritic cell's name reflects its distinctive morphology: long, thin cytoplasmic extensions, the dendrites, that spread out from the cell body like the branches of a tree, maximizing the surface area available for sampling the surrounding tissue. In the skin, the gut wall, the lung epithelium, and virtually every other tissue exposed to the environment, dendritic cells rest in a state of constant vigilance, extending their dendrites, engulfing and sampling the molecular contents of the tissue, detecting the molecular signatures of pathogens and cellular damage through their pattern recognition receptors. When they encounter foreign antigen, something remarkable happens. The dendritic cell transforms. It stops its tissue sampling, processes the engulfed material into peptide fragments, loads these fragments onto MHC molecules on its surface, and begins migrating via the afferent lymphatic vessels toward the regional lymph node. This migration, which takes approximately 12 to 24 hours, carries the antigenic evidence from the tissue where the threat was detected to the lymph node where the T cells that can recognize and respond to it are concentrated. In the lymph node, the mature dendritic cell presents its peptide MHC complexes to the thousands of naive T cells that percolate through the node, searching for the rare T cell, perhaps one in a million, whose T cell receptor recognizes the presented antigen. When it finds that T cell, it activates it, initiating the clonal expansion and differentiation that constitutes the adaptive immune response. The germinal center, the transient, highly organized microstructure that forms within the primary lymphoid follicles of lymph nodes and the spleen following B-cell activation, is one of the most remarkable biological structures in the body, generating antibody diversity through somatic hypermutation and selecting for high-affinity antibodies through a process that combines evolution and selection within the span of a single immune response. The germinal center has been called an evolutionary process in miniature, and the analogy is apt in ways that go beyond metaphor. It is a structure that appears, does its extraordinary work, and disappears within 3 to 4 weeks, existing only for the duration of the immune response that creates it and leaving behind its products, the high-affinity memory B cells and long-lived plasma cells, as its lasting contribution to immunological memory. When a B cell is activated by antigen binding to its surface receptor, and receives the necessary co-stimulatory signals from CD4 T helper cells, it migrates into the primary follicle and begins to divide rapidly. The dividing B cells form the germinal center, which reaches its maximum size of several thousand cells within about 2 weeks. Within the germinal center, B cells undergo a process unique in the genome's normal operation, somatic hypermutation. The activation-induced cytidine deaminase enzyme introduces random point mutations specifically into the variable regions of the immunoglobulin genes that encode the antibody's antigen-binding domains, at a rate approximately 1 million times the normal rate of DNA mutation. These mutations alter the amino acid sequence of the antibody's variable regions, and therefore the antibody's affinity for its target antigen. Some mutations will improve affinity, others will reduce it or abolish it entirely. The germinal center's selection process then acts on this mutational diversity. B cells that express antibodies with increased affinity for the antigen, which is retained in the germinal center in small amounts on the surface of follicular dendritic cells, receive survival signals and continue to proliferate. B cells with reduced affinity die by apoptosis. Over multiple rounds of mutation and selection, the average affinity of the germinal center's B cell population for the antigen increases, a process called affinity maturation that produces, after 3 to 4 weeks, antibodies with affinities sometimes 10 to 100 times higher than the original, pre-germinal center antibodies. Lymphedema. The swelling caused by the failure of the lymphatic system to adequately drain the interstitial fluid of the affected region is one of the most clinically significant consequences of lymphatic dysfunction and a condition affecting approximately 250 million people worldwide, with causes ranging from parasitic infection to surgical lymph node removal. The experience of lymphedema, the chronic, progressive swelling that results when the lymphatic system can no longer drain the tissues it is supposed to drain, is one of the clearest demonstrations of what the lymphatic system does when it works, and one of the most difficult chronic conditions to manage when it does not. Lymphedema most commonly affects a limb, an arm or a leg, when the lymphatic drainage of that limb has been disrupted by injury, infection, surgery, or radiation. The limb swells with the protein-rich fluid that the lymphatic capillaries have collected but cannot move forward to the venous circulation. Initially, the swelling is soft and pitting. Pressing a finger into the swollen tissue leaves a temporary indentation. Over time, the accumulated protein in the interstitial space stimulates inflammation and the progressive deposition of fibrotic tissue, and the swelling becomes firmer, harder, and less responsive to compression. The skin thickens, developing the characteristic hyperkeratosis and papillomatosis of chronic lymphedema. The risk of skin infection, erysipelas and cellulitis, increases dramatically because the protein-rich fluid in the swollen tissue provides an ideal culture medium for bacteria, and the impaired lymphatic flow reduces the supply of the immune cells that would normally clear bacterial invasion. Each infection further damages the remaining lymphatic vessels, perpetuating a cycle of deterioration. The most common cause of lymphedema worldwide is lymphatic filariasis, the infection of the lymphatic vessels by the parasitic nematodes Wuchereria bancrofti and Brugia malayi, transmitted by mosquito bites, which affects over 120 million people in the tropical and subtropical regions of Africa, Asia, and South America. In high-income countries, the most common cause is secondary lymphedema following cancer treatment, the surgical removal of lymph nodes and the radiation damage to lymphatic vessels that are consequences of breast cancer, pelvic cancer, and melanoma treatment. The macrophage, derived from monocytes that emigrate from the blood into the tissues, is the lymphatic system's primary phagocyte, engulfing and digesting cellular debris, foreign particles, and pathogens in the lymph nodes, the spleen, the liver, and virtually every other tissue of the body. and simultaneously serving as a sentinel that alerts the adaptive immune system to the nature of what it has consumed. The macrophage is one of biology's most versatile cells, a large, irregularly shaped phagocyte capable of engulfing particles up to its own size, of releasing a bewildering array of inflammatory and anti-inflammatory mediators, of presenting antigen to T cells, of directing the tissue repair that follows injury, and of performing very different functions in different tissue environments. The name comes from the Greek for large eater, which accurately describes the cell's defining capability but misses the complexity of what happens after the eating. A macrophage that engulfs a bacterium does not simply destroy it passively. It processes the bacterial proteins into peptide fragments, loads those fragments onto MHC class II molecules on its surface, and presents them to CD4 T helper cells in the same way that dendritic cells do. This antigen presentation activates the T helper cell, which in turn activates the macrophage more completely. A positive feedback loop that amplifies the macrophage's killing capacity and the inflammatory response. Macrophages also release the cytokines that initiate and coordinate the inflammatory response: tumor necrosis factor, interleukin-1, interleukin-6, the signaling molecules that produce fever, attract neutrophils to the site of infection, and stimulate the liver to produce the acute phase proteins of the systemic inflammatory response. In the lymph nodes, specialized macrophages in the subcapsular sinus capture pathogens and particulate antigens from the incoming lymph and transfer them to follicular B cells, bridging the first-line innate immune response with the antigen-specific adaptive response. In the liver, the Kupffer cells, the resident hepatic macrophages, filter the portal blood arriving from the gut, clearing the bacteria and bacterial products that cross the intestinal barrier even in health. The chyle, the milky, fat-laden lymph produced in the intestinal lacteals following a meal, contains the dietary fats, fat-soluble vitamins, and cholesterol absorbed from the gut in the form of large lipoprotein particles called chylomicrons, and the lacteal system of the small intestine is the primary route by which these lipids reach the systemic circulation. The appearance of chyle is startling when you first encounter it. Not the pale, watery liquid of most lymph, but a thick, milky white fluid that looks, frankly, like cream. This whiteness is not due to white cells or protein, but to the enormous numbers of chylomicron particles suspended in the fluid. Each chylomicron a sphere of triglyceride, phospholipid, and cholesterol approximately 100 to 500 nanometers in diameter. Too large to enter the blood capillaries of the intestinal villi, but small enough to squeeze through the large pores of the lacteals. The lacteals are the lymphatic capillaries of the small intestinal villi. Each villus, the tiny finger-like projection of intestinal mucosa that covers the inner surface of the small intestine, contains a single central lacteal that drains the fat-laden interstitial fluid of the villus into the collecting lymphatics of the intestinal wall. After a fatty meal, the lacteals fill with chylomicrons and the lymph becomes white, draining via the mesenteric lymphatics into the cisterna chyli and thence up the thoracic duct to empty into the venous blood at the left venous angle. The fat-soluble vitamins, A, D, E, and K are absorbed alongside the dietary fats as part of the chylomicron cargo, traveling with the chylomicrons through the lymphatic system rather than through the portal vein. This is why fat malabsorption, from any cause that impairs chylomicron formation or lacteal drainage, produces deficiency of the fat-soluble vitamins alongside the more obvious consequences of fat malabsorption. The lymphatic system is not merely an immune highway. It is, 3 times a day for most people, the delivery system for the essential nutrients that the diet provides. Natural killer cells, the lymphocytes that patrol the body without the antigen-specific receptors of T and B cells, kill virus-infected and tumor cells through the recognition of missing or altered MHC molecules on the target cell's surface, operating on the principle that normal, healthy cells express MHC class I and cells that have lost or altered this expression have been compromised. The natural killer cell is the lymphatic system's unconventional soldier, a cell that kills without needing to be educated in the thymus, without needing to learn a specific antigen, without the clonal selection that characterizes the adaptive immune response. Where T cells require prior exposure to an antigen before they can respond, natural killer cells are ready from birth, killing without prior sensitization, hence the name. The principle by which they operate is elegant and counterintuitive, rather than recognizing what a target cell has: a foreign antigen, a pathogen. A natural killer cell is triggered to kill by what a target cell lacks. Normal, healthy cells express MHC class I molecules on their surface, the antigen-presenting molecules that allow cytotoxic T cells to recognize and kill them if they become infected. Many viruses, to evade cytotoxic T cell killing, downregulate or eliminate MHC class I expression from the infected cell's surface. Cancer cells, similarly, often lose MHC class I expression as a mechanism of immune evasion. Natural killer cells have inhibitory receptors, primarily KIR receptors and NKG2A, that bind to normal MHC class I molecules and prevent the NK cell from killing the target. When a target cell has lost MHC class I expression, the inhibitory signal is absent, and the NK cell receives the activating signals from stress ligands expressed on infected or transformed cells through its activating receptors, and kills. The missing self hypothesis, the recognition that NK cells kill what lacks the normal self marker, provides a complementary mechanism to the cytotoxic T cells killing of what displays a foreign marker. Together, they close the immune evasion loop. The follicular dendritic cell, a non-hematopoietic cell resident in the germinal centers of lymphoid follicles, retains intact antigen on its surface for extended periods through complement receptors and Fc receptors, providing the antigenic stimulus against which germinal center B cells are selected for affinity maturation. The follicular dendritic cell is one of the more enigmatic cells of the lymphatic immune system, easily confused with the dendritic cells described earlier but, in fact, entirely unrelated to them in origin and mechanistically distinct in function. Where the conventional dendritic cell is hematopoietic, Derived from bone marrow precursors, the follicular dendritic cell is a resident stromal cell of the lymphoid follicle, of mesenchymal origin, that does not present antigen to T cells and does not itself have phagocytic activity. Its function is more specialized and in some ways more remarkable. It captures antigen-antibody complexes and complement-coded antigen from the lymph through its complement receptors and Fc receptors and holds them intact on its surface, not degrading them, but displaying them, for weeks to months. This retained antigen is the template against which the germinal center B cells are measured during affinity maturation. As B cells undergo somatic hypermutation and their antibody affinities change, they repeatedly test themselves against the antigen held on the follicular dendritic cell surface. Those B cells with higher affinity antibodies successfully compete for the limited antigen, receive the survival signals that allow them to continue, and proceed to further rounds of mutation and selection. Those with lower affinity fail to capture sufficient antigen and die. The follicular dendritic cell is, in this picture, the examiner, the constant reference point against which the evolving B cell repertoire is continuously tested. Without it, affinity maturation cannot proceed, and the immune response cannot generate the high-affinity antibodies that provide durable protection. Lymphatic filariasis, caused by the parasitic nematodes Wuchereria bancrofti, Brugia malayi, and Brugia timori, is the world's most common cause of lymphedema and one of the leading causes of disability in tropical countries, with the adult worms residing in the lymphatic vessels and lymph nodes where their presence and death trigger the fibrotic response that destroys lymphatic function. The life cycle of the filarial nematode that causes lymphatic filariasis is one of the more intimate parasitic relationships in medicine, a partnership between a mosquito vector, a human host, and a worm that makes its home in the lymphatic vessels, feeding on the lymph and reproducing for years within the body of an infected person. The adult worms, approximately 2 to 10 centimeters long, thread-like and barely visible to the naked eye, reside primarily in the afferent lymphatic vessels and lymph node sinuses, where they live for 6 to 8 years, continuously producing microfilariae, microscopic larval worms that circulate in the blood. The microfilariae exhibit a remarkable nocturnal periodicity in most populations, concentrating in the peripheral blood during the night, the hours when the Culex mosquito vectors are feeding, and retreating to the pulmonary capillaries during the day. When a mosquito feeds on an infected person, it ingests microfilariae with the blood meal, and the microfilariae develop through larval stages in the flight muscles of the mosquito before migrating to the mosquito's mouthparts, from which they are transmitted to a new human host during the next blood meal. The damage to the lymphatic system in filariasis is caused both by the living worms and by the host's immune response to them, and paradoxically, by the death of the worms after treatment. The inflammatory response to dead and dying adult worms can trigger the acute filarial lymphangitis, the painful, episodic attacks of fever, skin redness, and lymph node tenderness that represent the acute phase of the disease. And chronic exposure to these inflammatory episodes progressively scars the lymphatic vessels, reducing their function and eventually producing the irreversible lymphedema that is the disease's defining chronic manifestation. The mucosal immune system, the immune tissue associated with the mucous membranes of the gut, the respiratory tract, and the urogenital tract, is the body's largest immune compartment, containing approximately 70% of the immune cells in the body, and is organized into specialized lymphoid structures including the Peyer's patches of the small intestine and the bronchus-associated lymphoid tissue of the lung. The mucosal surfaces of the body, the inner linings of the digestive tract, the respiratory tract, and the urogenital system, are the body's most vulnerable frontiers, the places where the external world most intimately contacts the internal one. Across the entire gastrointestinal tract, the single-cell thick epithelial layer is all that stands between the trillions of bacteria of the gut microbiome and the sterile interior. The mucosal immune system, the diffuse lymphoid tissue distributed throughout these surfaces and the organized lymphoid structures embedded within them, is the specialized immune apparatus that manages this extraordinary interface. The Peyer's patches of the small intestine, named for the 17th century Swiss anatomist Johann Peyer, are the most prominent organized mucosal lymphoid structures, visible as raised oval patches on the antimesenteric wall of the small intestine, more numerous in the ileum and conspicuous in childhood, less so in adults. Each Peyer's patch is covered by a specialized epithelium containing M cells, the microfold cells whose name reflects their irregular apical surface, whose function is to transcytose antigens in microorganisms from the intestinal lumen to the underlying lymphoid tissue, delivering samples of the gut contents to the dendritic cells and macrophages below for immune surveillance. Within the Peyer's patch, B cells and germinal centers produce IgA, the predominant antibody class of mucosal immunity, which is then transported across the epithelium into the intestinal lumen, where it binds to microorganisms and prevents their adherence to the epithelial surface. The mucosal immune system maintains the delicate balance of the gut, tolerating the beneficial microbiome while remaining alert to pathogens, through mechanisms that are more complex and more precisely calibrated than any other aspect of the immune system. The complement system, a group of approximately 30 plasma proteins that circulate in the blood and lymph in inactive forms and are activated in a cascade by pathogen surfaces, antibodies, or cell damage, amplifies the innate immune response by directly lysing bacteria, coding pathogens for phagocytosis, and attracting immune cells to sites of infection through chemotactic fragments. The complement system is the lymphatic immune system's chemical amplification arm, a cascade of enzymatic reactions that, once initiated, generates an explosive local concentration of immune effector molecules. The system can be activated through three distinct pathways. The classical pathway, initiated by antibody bound to antigen, the lectin pathway, initiated by the binding of mannose-binding lectin or ficolins to carbohydrate patterns on pathogen surfaces, and the alternative pathway, initiated by the spontaneous hydrolysis of complement component C3 and its deposition on any surface that lacks the complement regulatory proteins found on normal host cells. All three pathways converge on the generation of the C3 convertase, an enzyme that cleaves C3, the most abundant complement protein in the plasma, into C3b, which covalently attaches to the pathogen surface, opsonization, and C3a, a small peptide fragment that acts as an anaphylatoxin. C3b on the pathogen surface serves as an opsonin, a chemical tag that phagocytes recognize through complement receptors, dramatically increasing the efficiency of phagocytosis. The complement cascade continues from C3 to C5, generating C5a, the most potent complement anaphylatoxin, a powerful attractant for neutrophils and monocytes that also activates mast cells and increases vascular permeability. Finally, C5b together with C6, C7, C8, and multiple copies of C9 assembles the membrane attack complex, a ring-shaped pore in the pathogen's lipid bilayer that allows water and ions to flow freely, lysing the organism. The entire cascade, from initial activation to membrane attack complex assembly, can be completed in seconds. The bone marrow, the primary lymphoid organ in which B cells develop and from which hematopoietic stem cells produce all the cellular elements of the blood and lymphatic immune system, is a soft, vascular tissue filling the medullary cavities of the flat bones and the ends of the long bones, producing approximately 500 billion cells per day to maintain the body's blood and immune cell populations. The bone marrow is the foundation of the entire immune system, the tissue from which every immune cell originates, where the lymphocyte precursors are born and where B cells complete their education. The hematopoietic stem cell, the undifferentiated progenitor that gives rise to all blood and immune cell lineages through a process of progressive differentiation commitment, resides in specialized niches within the bone marrow, supported by the stromal cells, extracellular matrix proteins, and cytokines that constitute the hematopoietic microenvironment. From the hematopoietic stem cell, two broad lineages arise: the common myeloid progenitor, which gives rise to red blood cells, platelets, neutrophils, eosinophils, basophils, monocytes, and mast cells, and the common lymphoid progenitor, which gives rise to T cells, B cells, natural killer cells, and innate lymphoid cells. B cells complete most of their development in the bone marrow. The pro-B cell rearranges its immunoglobulin genes. The pre-B cell expresses the pre-B cell receptor and undergoes selection, and the immature B cell expresses the mature B cell receptor and undergoes negative selection against self-antigens before being released as a naive mature B cell into the circulation. T cell precursors are exported from the bone marrow at an early stage and migrate to the thymus, where the thymic educational process described earlier completes their development. The daily cellular output of the bone marrow is staggering: 500 billion cells per day, every day, replacing the cells that are continuously dying or being consumed by the immune response. Red cells living 120 days, neutrophils 8 to 12 hours, platelets 10 days. The cytokines, the small, secreted signaling proteins that mediate communication between immune cells and between the immune system and other body systems, are the molecular language in which the lymphatic immune system conducts its business. coordinating cell activation, migration, proliferation, and death in the complex choreography of the immune response. Cytokines are the immune system's words, the molecular vocabulary through which immune cells communicate their state, their needs, and their instructions to each other and to the body as a whole. There are approximately 150 known cytokines, classified into several families. The interleukins, which originally referred to proteins that mediated communication between leukocytes, the interferons, originally characterized by their ability to interfere with viral replication, the tumor necrosis factors, the colony-stimulating factors that regulate hematopoiesis, the chemokines that direct cell migration, and the transforming growth factors that regulate cell proliferation and differentiation. Each cytokine binds to specific receptors on its target cells and triggers distinct signal transduction pathways that alter the cell's gene expression, metabolism, and behavior. Interleukin-2, produced by activated T cells, drives the proliferation of T cells. It is the growth factor that allows the clonal expansion essential for an adaptive immune response. Interleukin-4 and interleukin-13, produced by CD4 T helper cells, drive B cells toward IgA class switching, promoting the allergic and anti-parasitic immune responses. Interleukin-12, produced by dendritic cells and macrophages in response to bacterial infections, drives T cells toward the Th1 phenotype that promotes cellular immunity. Interferon gamma, produced by T cells and NK cells, activates macrophages to kill intracellular pathogens more effectively. Tumor necrosis factor, produced by macrophages in T cells in response to infection, drives inflammation, fever, and the systemic acute phase response. The cytokine network is not a simple hierarchy of command but a complex web of interactions in which the same cytokine may have different and sometimes opposing effects on different cell types in different contexts. A web that the immune system navigates in real time, integrating multiple simultaneous signals to produce the calibrated responses that protect without causing excessive collateral damage. The lymphatic valves, the bicuspid, semilunar valves distributed at intervals of 2 to 3 millimeters throughout the collecting lymphatic vessels, ensure that lymph flows in only one direction by opening when proximal pressure exceeds distal pressure and closing when flow would reverse, functioning analogously to the venous valves of the cardiovascular system. The lymphatic valves are the structural feature that transforms the collecting lymphatic vessels from simple tubes into directed conduits, preventing the backflow of lymph that would otherwise accompany the rhythmic contractions of the lymphangion and the intermittent compression of the surrounding skeletal muscles. Each valve consists of 2 semilunar leaflets of endothelial cells on a thin connective tissue stalk, projecting into the lumen of the vessel from opposite walls and meeting at their free edges when the valve is closed. The leaflets are oriented so that they open when the pressure in the proximal segment of the vessel exceeds that in the distal segment, as occurs when the proximal lymphangion contracts or when the surrounding tissue is compressed, allowing lymph to flow forward. When the pressure gradient reverses, as it does between the contractions of the lymphangion, The leaflets are pushed together by the reversed pressure differential and the valve closes, preventing backflow. The density of the valves, 1 every 2 to 3 millimeters, ensures that no segment of the collecting vessel is large enough to accumulate significant lymph between contractions, maintaining a steady directional flow despite the intermittent, non-synchronized contractions of successive lymphangins. In humans, lymphatic valve insufficiency, the failure of the valves to close adequately, allowing lymph to reflux, is one of the causes of primary lymphedema and is associated with mutations in the FOXC2 gene, which is required for the normal development of lymphatic valve leaflets. Immunological memory, the property of the adaptive immune system that enables faster, stronger, and more effective responses to pathogens encountered previously, is maintained by long-lived memory B cells and memory T cells that persist in the lymph nodes and bone marrow for decades, sometimes for the lifetime of the individual. The second encounter with a pathogen that has previously triggered an immune response is a fundamentally different experience for the immune system than the first encounter. Where the primary response takes 1 to 2 weeks to develop antibody production and cellular immunity, the secondary response generates high levels of high-affinity antibody within 2 to 3 days. Where the primary response produces antibodies of modest affinity, the secondary response produces the high-affinity antibodies refined by the germinal center's somatic hypermutation and selection. The cellular basis of this accelerated, enhanced secondary response is immunological memory, the long-lived population of antigen-experienced lymphocytes that persists after the primary response has resolved. Memory B cells, generated in the germinal center alongside the plasma cells that produce antibodies in the primary response, are quiescent, long-lived cells that circulate through the blood and lymph nodes, expressing the high-affinity antibody that the germinal center selected, ready to rapidly proliferate and differentiate into plasma cells upon re-exposure to their antigen. Memory T cells similarly persist after the primary response, circulating through the blood, the lymphoid organs, and the tissues, maintaining higher numbers than the naive T cells that preceded them, and responding to antigen re-exposure with faster expansion, lower activation threshold, and more rapid effector function. The durability of immunological memory is one of the immune system's most impressive properties. Measurable antibody responses to some childhood infections persist for 60 to 70 years, and vaccination against yellow fever, given as a single dose, provides protection that lasts decades or perhaps a lifetime. The memory cells established by the initial exposure or vaccination continue to maintain their numbers through homeostatic proliferation, driven by survival signals from the immune microenvironment even in the absence of antigen. The spleen's red pulp macrophages perform a critical role in iron recycling, capturing the hemoglobin released from destroyed red blood cells, breaking down the heme group to release the iron, and returning it to the circulation via transferrin for reuse in the synthesis of new hemoglobin in the bone marrow, a recycling process that conserves approximately 25 milligrams of iron per day. Iron is one of the most precious resources in the body, essential for hemoglobin synthesis, for mitochondrial respiration, for DNA synthesis, and for the function of dozens of enzymes. The daily dietary intake of iron, typically 10 to 20 milligrams, of which only 1 to 2 milligrams is actually absorbed, is far too small to sustain the production of hemoglobin for the 200 billion new red blood cells that must be produced every day. The solution is recycling. The iron released from the hemoglobin of senescent red cells destroyed in the spleen is nearly entirely recaptured and returned to the erythroid precursors in the bone marrow. The process begins when aging red cells, having lost the membrane flexibility required to squeeze through the narrow slits of the splenic sinuses, are trapped and phagocytosed by the red pulp macrophages. Within the macrophage, the hemoglobin is digested. The heme group is cleaved by hemoxygenase into iron, carbon monoxide, and biliverdin. The free iron is bound to ferritin, the iron storage protein, within the macrophage, and then exported via the ferroportin transporter on the macrophage surface, where it is captured by the plasma protein transferrin and delivered to the bone marrow for incorporation into new hemoglobin. Biliverdin is reduced to bilirubin, which is transported to the liver for conjugation and excretion in bile. The carbon monoxide, though seemingly inert, serves as a gaseous signaling molecule with vasodilatory and anti-inflammatory effects. The entire process recycles approximately 25 milligrams of iron per day, 20 to 30 times the daily dietary absorption, demonstrating the extraordinary efficiency of the body's internal iron economy. The structure of the antibody molecule: 2 heavy chains and 2 light chains arranged in a Y shape, with the constant regions providing structural and effector functions and the variable regions at the tips of the Y providing the unique antigen binding specificity of each antibody clone, is the molecular basis for both the extraordinary diversity of the immune repertoire and the multiple effector functions that different antibody classes mediate. The antibody molecule is one of biology's most elegant molecular designs. A Y-shaped glycoprotein of approximately 150 kilodaltons whose structure economically encodes both specificity and function in the same molecule. The 2 heavy chains and 2 light chains are connected by disulfide bonds, and each chain consists of a series of immunoglobulin domains, structurally similar beta sandwich folds that are the structural unit of the entire immunoglobulin superfamily. At the tips of the Y, in the Fab regions, each consisting of 1 variable heavy chain domain and 1 variable light chain domain folded against each other, are the antigen binding sites. The 6 hypervariable loops, 3 from the heavy chain variable domain and 3 from the light chain variable domain, come together to form the binding pocket whose precise shape and charge distribution determines what antigen the antibody will recognize. The extraordinary diversity of antibody specificities. The immune system can theoretically generate antibodies against perhaps 10 to the power of 15 different antigens, arises from the combinatorial assembly of antibody genes from multiple gene segments during B-cell development, the random selection of 1V, 1D, and 1J gene segment from the immunoglobulin heavy chain locus, the imprecise junction between these segments, the random pairing of heavy and light chains, and the somatic hypermutation of the germinal center together generate the nearly unlimited diversity that allows the immune system to respond to any antigen it might encounter. The constant regions of the heavy chains, forming the stem of the Y, determine the antibody class IgM, IgG, IgA, IgA, or IgD, and therefore the effector functions, Fc receptor binding for phagocyte-mediated killing, complement activation, placental transfer, or mast cell activation. Regulatory T cells, the CD4 T cells that express the transcription factor FOXP3 and suppress the activity of other immune cells, are the lymphatic system's internal peacekeepers, preventing the excessive inflammation, autoimmunity, and immune responses to harmless environmental antigens that would otherwise follow from the activity of the effector T cells. The immune system, capable of remarkable power and specificity in its response to threats, must also be equally capable of restraint, of knowing when not to respond, of limiting the collateral damage of inflammation, of tolerating the body's own tissues and the harmless microorganisms and dietary antigens that it encounters daily. This restraint is primarily provided by the regulatory T cells, a specialized population of CD4 T cells that express the master transcription factor FOXP3 and that suppress the activation and effector function of other T cells, B cells, and NK cells through contact-dependent mechanisms and through the secretion of anti-inflammatory cytokines including interleukin-10 and transforming growth factor beta. Regulatory T cells develop from two sources, in the thymus, where strong self reactivity in the negative selection process can lead not to deletion but to the diversion of the cell toward the regulatory lineage, the central trach, and in the periphery, where naive T cells encountering antigen and tolerogenic conditions, in the gut, in the presence of TGF-beta, can be converted to the regulatory phenotype, the peripheral trach. The importance of regulatory T cells for immune homeostasis is demonstrated catastrophically by their absence, the IPEX syndrome, immune dysregulation, polyendocrinopathy, enteropathy, X-linked, caused by mutations in the FOXP3 gene, produces a devastating multi-organ autoimmune disease in affected infants, with inflammatory bowel disease, type 1 diabetes, thyroiditis, and skin inflammation developing in the first weeks of life from the unrestrained activity of autoreactive T cells. The regulatory T cell is, in this sense, the immune system's check on its own power, the internal governance mechanism that prevents the immune response from destroying the very body it is trying to protect. The lymph node's architecture, with its capsule, cortex, paracortex, and medulla each housing different immune cell populations in spatially distinct zones, is maintained by a network of stromal cells that include the fibroblastic reticular cells, whose collagen-rich conduit system delivers small antigens rapidly from the afferent lymphatic sinus throughout the node. The lymph node's spatial organization is not incidental. It reflects the functional requirements of the immune response, which depends on specific cell types finding each other efficiently in a structure that may be filtering thousands of different antigens simultaneously. The fibroblastic reticular cells, the mesenchymal stromal cells that form the structural scaffold of the T-cell zone, produce and maintain a network of collagen I and ovalbumin conduits covered by their own cellular extensions. This conduit network is continuous with the subcapsular sinus and carries small antigens, molecules below approximately 70 kilodaltons in size, from the incoming lymph deep into the paracortex, where they can be sampled by the dendritic cells that survey the conduits and by the T cells that migrate through the FRC network. Larger antigens and particulate matter cannot enter the conduits and instead remain in the subcapsular sinus, where subcapsular sinus macrophages capture them. This size-dependent routing separates small soluble antigens, the kind most efficiently presented by dendritic cells to T cells, from larger particles and pathogens, the kind most efficiently captured by macrophages, and delivers each type to the cellular population best equipped to handle it. The FRC network also produces the chemokines CCL19 and CCL21 that attract naive T cells via their CCR7 receptor, maintaining the high density of T cells in the paracortex that ensures that the rare T cell with the matching T cell receptor will encounter the dendritic cell presenting its cognate antigen within the timeframe required for an effective immune response. The phenomenon of clonal selection, the process by which an antigen selects and activates from the pre-existing repertoire of lymphocytes only those cells that express receptors matching the antigen structure, was the theoretical achievement that explained how the adaptive immune system generates specificity without prior knowledge of what antigens it will encounter. The clonal selection theory, proposed by Frank McFarlane Burnett in 1957 and by David Talmage and Niels Jerne in related formulations around the same time, resolved a central puzzle in immunology. How does the immune system generate antibodies that are precisely specific for antigens the body has never encountered before? The earlier instructional theory proposed that the antigen somehow served as a template that instructed the antibody molecule to fold into the complementary shape. Burnett's clonal selection theory replaced this with an explanation that seemed almost too simple. The immune system pre-generates an enormous diversity of lymphocytes, each expressing a unique antigen receptor, before any antigen has been encountered. When an antigen enters the body, it selects, from this pre-existing repertoire, the lymphocytes whose receptors happen to match it, and those matching lymphocytes are triggered to proliferate, producing a large clone of cells all expressing the same specific receptor. The antibody or cellular response generated is therefore specific for the antigen because it is produced by a clone of cells selected by the antigen from a diverse pre-existing repertoire. The diversity of the repertoire is the key to the theory's success. If the repertoire is large enough to include at least one lymphocyte capable of binding any possible antigen, then the system can respond to any antigen it encounters. Burnett estimated the repertoire at perhaps 1 million different specificities. Current estimates place the number of distinct antibody specificities that the B-cell repertoire can generate at approximately 10 to the power of 11 or more. The spleen's white pulp responds to blood-borne antigens by generating germinal center reactions similar to those of the lymph node but without the afferent lymphatic input, instead sampling the blood directly through the marginal zone. a ring of specialized B cells that rapidly respond to T-independent antigens like bacterial polysaccharides. The marginal zone of the spleen, the region at the boundary between the white pulp and the red pulp, is home to a specialized population of B cells, the marginal zone B cells, that differ significantly from the follicular B cells that populate the primary follicles and participate in germinal center reactions. Marginal zone B cells are poised for rapid, T-independent responses to the polysaccharide antigens that coat the surface of encapsulated bacteria, Streptococcus pneumoniae, Neisseria meningitidis, Haemophilus influenza, that the conventional T-cell-dependent germinal center response is poorly suited to handle. T-independent antigens cannot be presented on MHC molecules to T cells, but many of them can directly cross-link multiple B cell receptors simultaneously, providing a sufficiently strong activating signal to drive B cell activation and antibody production without T cell help. Marginal zone B cells are particularly well suited to mounting these T-independent responses. They are located at the interface where the blood's contents are first examined by the splenic immune apparatus. They are pre-primed with a lower activation threshold than follicular B cells, and they can rapidly differentiate into plasma cells producing IgM antibody against the polysaccharide antigens within days of exposure. The clinical consequence of the loss of splenic marginal zone B cells, as occurs after splenectomy, is a dramatically increased susceptibility to infections with these encapsulated bacteria. The risk of overwhelming post-splenectomy infection, a rapidly fatal bacteremia that can progress from symptoms to death within hours, from Streptococcus pneumoniae and other encapsulated organisms is lifelong, and vaccinations against these organisms are given to every patient who undergoes splenectomy. The process of lymphangiogenesis, the formation of new lymphatic vessels, occurs during development, wound healing, and tumor progression, and is driven primarily by the growth factor VEGF-C acting on its receptor VEGFR3 on lymphatic endothelial cells, a pathway whose discovery has opened new avenues for therapeutic manipulation of lymphatic function. Lymphangiogenesis, the growth of new lymphatic vessels, is one of the body's responses to the increased fluid load and immune demands of tissue injury and inflammation. The primary driver of lymphatic vessel growth is vascular endothelial growth factor C, VEGF-C, a secreted growth factor produced by macrophages, mast cells, tumor cells, and the stromal cells of inflamed tissues. VEGF-C binds to the receptor VEGFR3, which is expressed almost exclusively on lymphatic endothelial cells in adult tissues, making it one of the most specific markers of the lymphatic endothelium. Binding of VEGF-C to VEGFR3 activates intracellular signaling cascades that promote lymphatic endothelial cell survival, proliferation, and migration, the three cellular processes required for the formation of new vessel sprouts. In wound healing, lymphangiogenesis is essential for the restoration of normal fluid drainage after the inflammation and edema of the acute inflammatory response. In tumor biology, lymphangiogenesis has taken on particular clinical significance. Many tumors produce VEGF-C, stimulating the growth of new lymphatic vessels within and around the tumor. And these new lymphatic vessels provide the route by which tumor cells travel to the regional lymph nodes in the earliest stage of lymph node metastasis. The detection of lymphatic invasion by tumor cells and the involvement of regional lymph nodes by metastases are among the most important prognostic factors in many cancers, making the VEGF-C-VEGFR3 signaling axis both a marker of disease stage and a potential therapeutic target for preventing lymphatic metastasis. You have been with me for a while now, traveling through the capillaries and the nodes and the germinal centers and the dendritic cells and the lymph that moves through it all. I find that genuinely wonderful. If these nights together mean something to you, if there is something in the pace of this, in the way the quiet science of the body becomes a kind of rest for the mind, then subscribing to Relax with Science is a simple and warm way to say, yes, I would like more of this. I would like to come back. And I will be here whenever you do. The lymphatic system's role in lipid absorption is so fundamental that people with primary lymphatic disorders, conditions that impair lymphatic vessel development or function, develop severe malabsorption of dietary fat and fat-soluble vitamins, protein-losing enteropathy, and chylous effusions in the pleural and peritoneal cavities. The clinical consequences of lymphatic dysfunction in the gut illustrate, with uncomfortable clarity, how central the lymphatic system is to normal nutrition. The lacteals of the intestinal villi are the sole route of absorption for dietary long-chain fatty acids and the fat-soluble vitamins A, D, E, and K. These molecules are too large and too lipophilic to enter the blood capillaries and must travel through the lymphatic system as chylomicrons. When the lacteals or their drainage pathways are obstructed or functionally impaired, by primary lymphatic dysplasia, by congenital intestinal lymphangiectasia, by the lymphatic damage of radiation therapy, or by the inflammatory obstruction of Crohn's disease, the chylomicrons cannot drain normally and the dietary fat is not effectively absorbed. The malabsorption of fat produces steatorrhea, the passage of fat in the stool, and the malabsorption of fat-soluble vitamins produces the deficiency diseases, vitamin A deficiency causing night blindness and immune impairment, vitamin D deficiency causing rickets and osteomalacia, vitamin E deficiency causing peripheral neuropathy, vitamin K deficiency causing impaired coagulation, and protein-losing enteropathy, a complication of intestinal lymphangiectasia in which the dilated, leaking lacteals weep albumin and other plasma proteins into the intestinal lumen. The plasma protein level falls dramatically, producing the edema of hypoalbuminemia, chylous ascites and chylothorax, the accumulation of milky, fat-laden lymph in the peritoneal and pleural cavities respectively, caused by the rupture or leak of major lymphatic vessels, are rare but dramatic manifestations of lymphatic injury that vividly illustrate what the lymphatic system normally prevents from going where it does not belong. The interferon response, the innate antiviral defense system in which virus-infected cells signal their neighbors to enter a state of heightened antiviral resistance, operates through secreted interferon proteins that activate a program of antiviral gene expression in surrounding cells, providing an early, non-specific defense while the adaptive immune response is being assembled. The interferons were among the first cytokines to be discovered, observed in 1957 by Alec Isaacs and Jean Lindenman as a factor produced by virus-infected cells that interfered with the replication of a second virus in adjacent cells. The type I interferons, primarily interferon alpha and interferon beta, are the primary antiviral interferons, produced by virtually any cell that detects the presence of viral nucleic acids through its innate immune pattern recognition receptors, the toll-like receptors that sample the endosomal compartment for foreign RNA and DNA, and the cytoplasmic sensors RIGI and CGAS that detect viral RNA and DNA in the cytoplasm. These pattern recognition receptors detect the molecular signatures of viral nucleic acid, double-stranded RNA, which is produced as an intermediate in the replication of many RNA viruses, unmethylated CpG DNA characteristic of certain viral genomes, and other molecular patterns absent from normal cellular nucleic acids. When a pattern recognition receptor detects a viral signature, the infected cell activates the transcription factor IRF3, which drives the transcription of interferon genes, and the cell secretes interferon into the surrounding tissue. Adjacent cells bind the secreted interferon through interferon receptors on their surface and activate the JAK-STAT signaling pathway, which drives the expression of several hundred interferon-stimulated genes. Genes encoding proteins that inhibit viral replication at multiple steps, preventing viral entry, degrading viral RNA, inhibiting viral protein synthesis, and activating NK cells that can kill infected cells. The interferon response provides the hours to days of innate antiviral protection that allow time for the adaptive immune response to develop, and is so effective that viruses have evolved dozens of mechanisms specifically to evade or suppress it. The anatomy of the lymph node, with its precisely organized zones directing specific immune cell populations to particular microenvironments, is maintained by the chemokine network, a system of attractant proteins that guide the migration of lymphocytes in dendritic cells to their correct locations within the node with a precision that is essential for immune function. The lymph node's organized structure, T cells in the paracortex, B cells in the follicles, dendritic cells concentrated at the TB boundary, macrophages in the sinuses, is not maintained by physical barriers between these zones but by the differential expression of chemokines and their receptors across the node's microenvironments. Naive T cells and mature dendritic cells express CCR7, the receptor for the chemokines CCL19 and CCL21, which are produced by the fibroblastic reticular cells of the T cell zone. The gradient of these chemokines, highest in the paracortex, attracts naive T cells and dendritic cells to the T cell zone, where their encounter is most likely to trigger T cell activation. Naive B cells express CXCR5, the receptor for CXCL13, which is produced by the follicular dendritic cells and stromal cells of the B cell follicles. The gradient of CXCL13, highest in the follicle, attracts naive B cells to the follicular zone, where their antigen sampling and activation occur. When T cells and B cells are activated and need to interact for the initiation of germinal center reactions, they both downregulate their homing receptors for their respective zones and upregulate the others. Activated T cells upregulate CXCR5 and move toward the follicle, while antigen-activated B cells upregulate CCR7 and move toward the T cell zone. They meet at the TB boundary, where the T cell provides the CD40L-mediated co-stimulatory signals and the cytokines that B cells need for germinal center entry. This chemokine choreographed dance of cellular positioning, repositioning, and directed migration is the spatial organization of immunity. The movement of the right cells to the right places at the right times. The process of T cell activation requires three distinct signals delivered simultaneously: antigen recognition through the T cell receptor, co-stimulation through molecules like CD28 binding to B7 on the antigen-presenting cell, and cytokine signals from the inflammatory environment. And the absence of any one signal produces energy rather than activation, a state of unresponsiveness that prevents immune responses to self-antigens. The three-signal model of T-cell activation is the immune system's safety mechanism against accidental autoreactivity. A T cell that receives only signal 1, antigen recognition through its T cell receptor, without the co-stimulatory signal 2 becomes anergic, entering a state of long-lived unresponsiveness to that specific antigen. This is the mechanism of peripheral tolerance. T cells that escape the thymic negative selection and enter the periphery-bearing receptors that could recognize self-antigens are exposed to self-antigens continuously, but without the co-stimulatory signals provided by activated antigen-presenting cells. They become anergic rather than activated. The co-stimulatory molecules, B7-1, CD80, and B7-2, CD86, on the antigen-presenting cell, binding CD28 on the T cell, are upregulated only on dendritic cells and macrophages that have been activated by the innate immune detection of pathogen-associated molecular patterns. Self-tissues that do not express these co-stimulatory molecules present self-antigen to T cells but do not provide co-stimulation, inducing anergy. The third signal, the cytokine context, determines the differentiation of the activated T cell. Interleukin-12 and interferon gamma promote Th1 differentiation towards cellular immunity. Interleukin-4 promotes Th2 differentiation toward humoral immunity and antiparasitic responses. Interleukin-6 and TGF-beta promote Th17 differentiation toward mucosal defense. TGF-beta alone promotes regulatory T cell differentiation. The complete immune response is thus not simply the recognition of antigen but the interpretation of antigen in a specific inflammatory context, with the consequences of misinterpretation, anergy, autoimmunity, or inappropriate effector function, providing the selection pressure that has made the three-signal requirement an evolutionarily conserved safety mechanism. The lymphatic system of the brain. The glymphatic system is a recently discovered network that uses the perivascular spaces surrounding the brain's arteries and veins as channels through which cerebrospinal fluid flows, flushing metabolic waste products including amyloid beta from the brain parenchyma, predominantly during sleep. The brain was long considered to be an exception to the rule of lymphatic drainage. The central nervous system was thought to lack the lymphatic vessels present in other tissues, and the removal of metabolic waste from the brain was attributed entirely to the diffusion of waste products into the cerebrospinal fluid and their clearance through the arachnoid villi. The discovery of the glymphatic system by Mike N. Nedergaard and colleagues in 2012 revealed a previously unsuspected waste clearance mechanism that has the character of a lymphatic function, even if it is not technically composed of lymphatic vessels. The system works through the perivascular spaces, the spaces surrounding the brain's arteries and veins, enclosed by the end feet of the astrocytes that ensheath the vessels. Cerebrospinal fluid, produced in the choroid plexus of the ventricles, flows along the perivascular spaces surrounding the arteries into the brain parenchyma, driven by the pulsatile expansion and contraction of the arteries with each heartbeat. Within the parenchyma, the CSF mixes with the interstitial fluid and its dissolved contents, including the metabolic waste products of neural activity, among them amyloid beta, the peptide whose aggregation is the defining pathological feature of Alzheimer's disease. The waste-laden interstitial fluid then drains along the perivascular spaces surrounding the veins, eventually flowing into the cervical lymph nodes. The glymphatic system is dramatically more active during sleep, when the extracellular space of the brain expands by approximately 60%, increasing the efficiency of the flush. The finding that sleep deprivation impairs glymphatic clearance and increases brain amyloid beta levels provides a direct mechanistic link between poor sleep and the risk of Alzheimer's disease, and it provides one of the most compelling biological arguments for the importance of adequate sleep. The gut-associated lymphoid tissue, by far the largest component of the mucosal immune system, must simultaneously maintain tolerance to the trillions of bacteria of the gut microbiome, respond to genuine pathogens when they breach the epithelial barrier, and generate the secretory IgA that is the first line of mucosal antibody defense. The gut is the most immunologically complex environment in the body, an interface where the immune system must perform what might seem like contradictory tasks simultaneously. On one side of the intestinal epithelium are the trillions of bacteria of the microbiome, beneficial organisms that collectively perform essential functions in nutrient metabolism and immune development, and that are present in numbers that would, in any other tissue, trigger a devastating inflammatory response. On the other side is the largest population of immune cells in the body, the lamina propria lymphocytes, the intraepithelial lymphocytes, the Peyer's patches and isolated lymphoid follicles, and the mesenteric lymph nodes, all of which must be maintained in a state of active surveillance without mounting the full-scale inflammatory response that the presence of bacteria would normally trigger. The resolution of this paradox is achieved through several mechanisms. The healthy gut epithelium presents an effective physical barrier through its tight junctions. The Treg cells of the lamina propria, stimulated by microbial metabolites including the short-chain fatty acids produced by commensal bacteria, maintain active immunological tolerance to the microbiome. The secretory IgA produced by the lamina propria plasma cells and transported across the epithelium into the lumen binds to microorganisms and neutralizes pathogens without triggering complement activation or inflammatory responses, an effector mechanism sometimes called immune exclusion. And the innate immune sensing of the epithelium is carefully calibrated to distinguish the routine presence of commensal bacteria from the breaching of the barrier by genuine pathogens. The balance of this immune microbiome relationship, maintained by the gut's lymphoid tissue, is one of the most consequential biological partnerships in the body. The lymphatic vessels of the skin, the cutaneous lymphatics, play a critical role in the drainage of fluid and immune cells from the skin and in the trafficking of cutaneous dendritic cells to the regional lymph nodes, and their dysfunction contributes to the chronic skin changes and increased infection risk of lymphedema affecting the extremities. The skin is the body's largest organ by surface area and one of its most immunologically active, constantly sampling the antigens, microorganisms, and irritants that the environment presents to its surface. The Langerhans cells of the epidermis, the skin-resident dendritic cells, survey this surface, capturing antigens and presenting them to T cells. When activated by the detection of foreign antigen, Langerhans cells migrate out of the epidermis into the dermal lymphatics, following the gradient of CCL19 and CCL21 produced by the lymphatic endothelial cells themselves. The lymphatic endothelial cells of the skin also express CCL21 on their luminal surface, binding to CCR7 on the emigrating dendritic cells and facilitating their entry into the lymphatic capillary. From there, the dendritic cells travel in the lymph to the regional lymph nodes, the axillary nodes for the arm and lateral chest, the inguinal nodes for the leg, where they present their captured cutaneous antigens to naive T cells. This dendritic cell trafficking is the mechanism by which contact hypersensitivity, the allergic reaction to substances like nickel, latex, and poison ivy, is initiated. The Langerhans cells capture the hapten from the skin, travel to the lymph node, and prime T cells that then become the effectors of the subsequent allergic reaction on re-exposure. In lymphedema, the impaired cutaneous lymphatic drainage reduces this dendritic cell trafficking, potentially contributing to the altered immune responses of chronic lymphoedematous skin. The T cell receptor's diversity, estimated at approximately 10 to the 18th power distinct specificities, is generated by the VDJ recombination process that assembles the receptor genes from multiple gene segments during T cell development in the thymus, and the random imprecision of this assembly creates a diversity that is practically unlimited relative to the antigenic universe. The T cell receptor's extraordinary diversity, the ability to recognize virtually any peptide MHC complex that might be generated by any pathogen or tumor, is generated by the same fundamental mechanism as antibody diversity, adapted for the T cell context. The alpha and beta chains of the T cell receptor are encoded by gene loci similar in organization to the immunoglobulin loci, multiple V, variable, D, diversity, for the beta chain, and J, joining, gene segments that are randomly selected and assembled during T-cell development by the RAG1/RAG2 recombinase. The combinatorial diversity from the random selection of gene segments provides perhaps 10 to the power of 6 distinct receptor configurations. But the junctional diversity added by the imprecise joining of gene segments, in which terminal nucleotides are removed and random nucleotides are added at the VD and DJ junctions by the enzyme TDT, dramatically amplifies this combinatorial diversity. The junctional sequences encode the CDR3 loops of the T-cell receptor, the loops that make the most direct contacts with the antigen, and their random generation creates the near-infinite diversity in antigen contact surfaces that allows the T-cell repertoire to respond to any antigen. The combination of combinatorial and junctional diversity in the alpha and beta chains, and the random pairing of these chains, is estimated to generate a theoretical diversity of approximately 10 to the 18th possible T-cell receptor specificities, a number that exceeds the number of T-cells in the body by many orders of magnitude, ensuring that the diversity of the receptor repertoire is never a limiting factor in the immune response to novel antigens. The phenomenon of linked recognition, the requirement that T cells and B cells responding to the same antigen must recognize epitopes on the same antigen molecule, even if the specific epitopes differ, is the molecular basis for the T-cell dependence of B-cell responses and for the immunogenicity of protein antigens relative to polysaccharides. The requirement for linked recognition is one of the most important rules governing the collaboration between T and B cells in the adaptive immune response. A B cell that recognizes and internalizes an antigen through its surface immunoglobulin processes the entire antigen into peptide fragments and presents them on MHC class II molecules to CD4 T helper cells. The T helper cell that receives this signal must be specific for a peptide derived from the same antigen molecule, the linked requirement. This linkage ensures that T cells and B cells engaged in producing a response to a given antigen are indeed working in the same direction, responding to the same molecular threat, rather than randomly cross-activating each other. The linked recognition requirement explains why protein antigens are T-dependent. They can be processed and presented to T cells as peptides, satisfying the linkage requirement, while polysaccharides are T-independent. They cannot be processed into peptides presentable on MHC class II and therefore cannot engage T helper cells. T-dependent antigens generate germinal center reactions, affinity maturation, class switching, and memory, the full range of adaptive immune tools. T-independent antigens generate primarily IgM antibody responses without affinity maturation or durable memory. This is why conjugate vaccines, which chemically attach polysaccharide antigens, the target, to protein carriers, the T-cell activator, produce T-dependent responses to polysaccharide antigens that the polysaccharide alone would not generate, producing the memory and class-switched responses that provide more durable protection. The lymphangin, the functional unit of the collecting lymphatic vessel, consisting of the segment of vessel between two consecutive valves, contracts spontaneously and rhythmically under the control of intrinsic pacemaker activity in the lymphatic smooth muscle, with contraction frequency and amplitude regulated by mechanical stretch, nitric oxide, histamine, and many other local mediators. The lymphangin is the lymphatic system's functional answer to the absence of a dedicated pump, each segment of collecting lymphatic vessel between two valves acting as an independent contractile unit, capable of spontaneous, rhythmic contraction that propels lymph forward. The smooth muscle cells of the lymphangin wall express ion channels that generate spontaneous membrane potential oscillations, the lymphatic pacemaker potential, at a frequency of approximately 5 to 20 contractions per minute under basal conditions. When the lymphangin is stretched by the accumulation of lymph within it, signaling that more lymph is arriving than is currently being propelled forward, the stretch increases both the frequency and the force of the contractions, accelerating lymph propulsion. This stretch-sensitive regulation creates an intrinsic feedback loop. High lymph formation rates produce lymphangin distension, which increases contraction frequency and force, which increases flow, which reduces distension toward its set point. Nitric oxide from the lymphatic endothelium acts as an inhibitory modulator, reducing contraction frequency when basal lymph flow is adequate, and histamine from mast cells acts as an activating modulator during inflammation, increasing contraction frequency when the inflammatory fluid load is high. The autonomic nervous system, sympathetic fibers innervating the smooth muscle, also modulates lymphangin contractility, providing a mechanism for the global upregulation of lymphatic pumping during exercise. The lymphangin is, in miniature, as sophisticated a pump as the heart, regulated by the same types of mechanical and chemical inputs, performing the same function at a smaller scale. Before we go further, feel the body right now. Not to look for anything, not to check anything. Simply feel the weight of the limbs, the warmth of the skin, the gentle movement of the breath. Somewhere in the tissues of this body, in the spaces between the cells, in the tiny lymphatic capillaries that thread through every organ, the lymph is moving. Slowly, quietly, without any sensation you can feel, the fluid is returning. The immune cells are circulating. The body is doing its quiet maintenance. You do not need to know it is happening. It is happening. Let the body rest in that. The phenomenon of original antigenic sin, the tendency of the immune system to preferentially respond to the antigens of the first pathogen strain encountered rather than the antigens of subsequent, antigenically related but distinct strains, was first described for influenza and has implications for vaccine design and the cross-reactive immunity of related pathogens. Original antigenic sin, named by Thomas Francis Jr. in 1960, who noticed that the antibody response to influenza infection in adults was dominated by antibodies against the influenza strains the person had encountered in childhood, is the memory B cell's conservatism in the face of antigenic variation. When the immune system has already established memory B cells against the particular antigen, A subsequent infection with an antigenically related but distinct pathogen strain activates those pre-existing memory cells preferentially, producing large amounts of antibody against the original antigen, even if the original antigen-specific antibody is less effective against the new variant than an antibody against the new variant's specific epitopes would be. The mechanism reflects the kinetics of immunological memory. Memory B cells, pre-activated and poised for rapid response, respond more quickly and with lower activation thresholds than the naive B cells that would generate a new, strain-specific response. The rapid activation of original memory cells outcompetes the slower priming of new naive cells, and the antibody response that dominates the secondary immune response is largely directed against the original strain's antigens. The clinical significance of this phenomenon extends beyond influenza. It may contribute to the variable efficacy of influenza vaccines in people who have had many prior influenza exposures, because their response to the new vaccine strain is dominated by their oldest immunological memories. And it may explain aspects of the cross-reactive immunity seen when related viral strains circulate. Why exposure to one coronavirus provides partial protection against another. For instance, because the original antigenic sin phenomenon can provide cross-reactive responses even against novel variants. The concept of immune surveillance, the continuous patrolling of the body's tissues by immune cells for the early detection and elimination of transformed cells that could become cancer, is the theoretical basis for the immunological control of cancer and explains both why the immune system can eliminate many early tumors and why cancers that develop must have evaded this surveillance. The immune surveillance hypothesis, first proposed by McFarlane Burnett and Lewis Thomas in the 1950s and 1960s, proposed that the immune system continuously monitors the body's cells for signs of malignant transformation and eliminates nascent cancer cells before they can form a clinically detectable tumor. The hypothesis was controversial for decades because tumors clearly do develop despite the intact immune system, suggesting either that the surveillance was incomplete or that tumors actively evaded it. The modern concept of cancer immunoediting, developed by Robert Schreiber and colleagues from the late 1990s, has largely resolved this tension by proposing three phases: elimination, equilibrium, and escape. In the elimination phase, the innate and adaptive immune system destroys transformed cells bearing tumor antigens. This process may be entirely successful, eliminating many incipient tumors before they can become clinically apparent. In the equilibrium phase, some tumor cells survive the elimination pressure but are held in a state of immune-mediated dormancy for years or decades, not eliminated but not able to progress, as the immune pressure matches the tumor's growth capacity. In the escape phase, tumor cell variants that have evaded immune recognition, by losing antigen expression, by downregulating MHC molecules, by expressing immune checkpoint ligands that suppress T-cell responses, emerge from immune control and progress to the clinically apparent tumors that present for treatment. The immune checkpoint immunotherapy that has transformed oncology, the PD-1, PD-L1, and CTLA-4 inhibitors, works precisely by restoring the immune surveillance function that the tumor had suppressed, allowing the patient's own immune system to recognize and destroy the cancer. The lymphatic endothelium expresses a distinct molecular signature from the vascular endothelium, including LYVE1, potoplanin, PROX1, and VEGFR3, that reflects its unique developmental origin from a subset of vascular endothelial cells in the cardinal vein, and that provides the molecular markers used to identify lymphatic vessels in tissue sections and to track their development. The molecular identity of the lymphatic endothelium was established largely through the work of researchers who needed to distinguish lymphatic from blood vascular endothelium in tissue sections, a distinction that is visually difficult under the light microscope, since lymphatic vessels and blood capillaries are both thin-walled, endothelium-lined tubes of similar dimensions. The discovery of markers specifically expressed by lymphatic endothelial cells, most importantly the transcription factor PROX1, now considered the master regulator of lymphatic endothelial identity, the hyaluronan receptor LYVE1, the membrane glycoprotein potoplanin, and the growth factor receptor VEGFR3, provided the immunohistochemical tools to specifically identify lymphatic vessels. These markers also revealed the developmental origin of the lymphatic endothelium. PROX1 expression was found in a subset of endothelial cells in the cardinal vein at approximately embryonic day 9.5 in the mouse, and the tracking of these PROX1 positive cells revealed that they bud off from the cardinal vein, the primary vein of the embryo, and migrate to form the lymph sacs from which the lymphatic vascular network develops. PROX1 acts as a fate-determining transcription factor that reprograms vascular endothelial cells toward the lymphatic endothelial program. Mice in which PROX1 is deleted lack lymphatic vessels entirely, demonstrating its essential role, while forced expression of PROX1 in blood vascular endothelial cells induces the expression of the lymphatic program. The molecular distinction between lymphatic and blood endothelium is maintained throughout life by the continued expression of PROX1, whose downregulation even in adult lymphatic endothelial cells causes the cells to revert toward a blood vascular gene expression pattern. The spleen's role in removing encapsulated bacteria from the bloodstream, through the opsin-independent phagocytosis of these organisms by the marginal zone macrophages and the rapid IgM production of the marginal zone B cells, makes the spleen irreplaceable in the defense against certain specific bacterial pathogens, particularly in young children whose adaptive immune repertoire is not yet fully developed. The spleen's unique contribution to antibacterial defense is not uniformly distributed across all bacterial pathogens but is specifically critical for the encapsulated bacteria, Streptococcus pneumoniae, Neisseria meningitidis, Haemophilus influenza, whose polysaccharide capsule provides the primary virulence mechanism by resisting phagocytosis. The polysaccharide capsule of these bacteria inhibits complement-mediated opsonization and prevents macrophages from efficiently phagocytosing the organisms. The specific anti-polysaccharide antibodies produced by the marginal zone B cells, and the complement deposition that these antibodies facilitate through the classical pathway, are the primary mechanism by which the spleen can efficiently clear these organisms from the blood. when the spleen is absent, from surgical removal following trauma, from functional asplenia in sickle cell disease, or from congenital asplenia, this specific defense is lost. The blood no longer receives the rapid marginal zone IgM response to newly encountered polysaccharide antigens, and the filtration and phagocytosis of opsonized capsulate bacteria in the splenic red pulp no longer occurs. The result is an increased susceptibility to overwhelming infection with these specific organisms that is lifelong, but most dangerous in young children, in the first years after splenectomy, and in people with functional asplenia from hemoglobin disorders. The post-splenectomy protocol, vaccination against the encapsulated organisms, prophylactic penicillin in young children, and the prescription of antibiotics for any febrile illness, is an attempt to compensate pharmacologically for the specific immunological function that the spleen provides and that no other organ can fully replace. The phenomenon of affinity maturation, the progressive increase in antibody affinity for antigen that occurs over the course of a germinal center reaction through somatic hypermutation and selection, can increase antibody affinity by 10 to 1,000-fold relative to the initial primary immune response antibody, and is the primary mechanism by which secondary immune responses are qualitatively superior to primary ones. The numbers of affinity maturation are remarkable. An initial B cell that escapes the bone marrow carries an antibody with an affinity for its target antigen, typically measured as the dissociation constant Kd, of approximately 10 to the -7 molar. After passage through the germinal center and multiple rounds of somatic hypermutation and selection, the antibody affinity can reach 10 to the -10 or 10 to the -11 molar, 1,000 to 10,000 times tighter binding to the antigen. The biological consequences of this affinity increase are profound. A high-affinity antibody can neutralize a virus at lower concentrations, providing protection even when the antibody level is low, as it is in the later months after vaccination. A high-affinity antibody remains bound to its antigen for longer. The half-life of the antigen-antibody complex is proportional to affinity, providing more effective opsonization and complement activation. And a high-affinity antibody that crosses the placenta from a vaccinated mother to her fetus provides more durable protection to the newborn than a lower-affinity antibody would. The progressive affinity maturation over the course of the germinal center reaction, which can continue for weeks after the initial infection or vaccination has resolved, means that the quality of the antibody response continues to improve even after the acute immune response is over. This progressive improvement is one of the reasons that the immune responses generated by natural infection or vaccination often continue to be refined for months, and why the timing and spacing of vaccine doses can affect the quality of the antibody response generated. The Peyer's patches and isolated lymphoid follicles of the small intestine are covered by the follicle-associated epithelium, which contains the M cells that transcytose antigens and microorganisms from the intestinal lumen across the epithelial barrier to the underlying lymphoid tissue, initiating the mucosal immune response to luminal contents. The M cell, microfold cell, is one of the lymphatic immune system's most specialized and least celebrated cellular inventions. It is an epithelial cell that has essentially abandoned the absorptive and secretory functions of the intestinal enterocyte in order to devote itself entirely to antigen sampling and delivery. The apical surface of the M cell, the surface facing the intestinal lumen, lacks the elaborate brush border of microvilli that covers the enterocyte. Instead, it has a sparse covering of irregular microfolds from which it takes its name, creating a larger surface area that facilitates the uptake of particulate material and microorganisms from the lumen. The basolateral surface of the M cell, facing the underlying lymphoid tissue, is deeply invaginated, forming a pocket into which dendritic cells, macrophages, and lymphocytes extend their processes, waiting to receive the antigens that the M cell has transcytosed from above. The M cell takes up a wide variety of antigens and microorganisms from the lumen by endocytosis and phagocytosis, and transports them across the cell in vesicles, releasing them into the subepithelial dome on the basolateral side, the dome-shaped region of the Peyer's patch immediately beneath the follicle-associated epithelium, where they are captured by dendritic cells and macrophages. The M cell pathway is exploited by some pathogens, Salmonella, Listeria, and poliovirus among them, that have evolved mechanisms to enter and cross M cells, using the transepithelial pathway that the M cell provides for antigen sampling as a route of invasion. The concept of pattern recognition, the detection of pathogens by the innate immune system through toll-like receptors, nod-like receptors, RIGI-like receptors, and other pattern recognition receptors that bind conserved molecular structures shared across broad classes of microorganisms, is the foundational principle of innate immunity and connects the lymphatic immune system to the deep evolutionary history of immunity. The toll-like receptors, the first family of pattern recognition receptors to be identified in mammals, following the characterization of the Drosophila toll protein by Jules Hoffman and the identification of its mammalian homologs by Bruce Beutler and Charles Janeway, are the innate immune system's ancestral detectors of microbial threat. The toll-like receptors detect the conserved molecular structures of microorganisms, the pathogen-associated molecular patterns, that are shared across broad taxonomic classes and that are structurally distinct from the molecules of mammalian cells. TLR4 detects lipopolysaccharide, the glycolipid component of the outer membrane of gram-negative bacteria. TLR5 detects bacterial flagellin, the protein subunit of bacterial flagella. TLR9 detects unmethylated CpG DNA, characteristic of bacterial and viral DNA. TLR3 detects double-stranded RNA, produced during viral replication. The crucial insight of pattern recognition theory, articulated by Charles Janeway in 1989 before the molecular identity of the receptors was known, is that the innate immune system does not recognize any specific pathogen but rather the molecular signatures that are common to entire classes of microorganisms and that are structurally impossible for pathogens to lose without compromising their fitness. The conserved nature of these molecular patterns, combined with the pattern recognition receptor's unchanging specificity, provides the innate immune system with a set of detectors that can reliably identify any microorganism as belonging to the dangerous category, foreign, microbial, without needing to have encountered the specific organism before. Hoffman, Beutler, and Steinman shared the Nobel Prize in 2011 for the discoveries of toll-like receptors in dendritic cells. The lymphatic system's development during embryogenesis follows a precise sequence beginning with the specification of lymphatic endothelial precursors in the cardinal vein at around embryonic day 9 in the mouse, and mutations in the transcription factors and growth factors that regulate this development produce the hereditary lymphedema syndromes. The embryological development of the lymphatic system is one of the more recently understood developmental processes. The molecular mechanisms governing it were largely unknown until the discovery of the key molecular players in the 1990s and 2000s. The process begins when a subset of endothelial cells in the anterior cardinal vein begin expressing the transcription factor PROX1, which reprograms them toward the lymphatic endothelial fate. These PROX1 positive cells then bud off from the cardinal vein, migrate, and coalesce to form the primary lymph sacs, the jugular lymph sacs in the neck region, from which the lymphatic vascular network progressively expands through the sprouting and branching of new lymphatic endothelial cells. The growth of the lymphatic network is driven primarily by the VEGF-C-VEGFR3 signaling axis. VEGF-C produced by the surrounding mesenchyme activates VEGFR3 on the lymphatic endothelial cells, promoting their migration and proliferation. Mutations in the genes encoding the key developmental proteins produce the hereditary lymphedema syndromes. Milroy disease, primary congenital lymphedema presenting at or shortly after birth with lower limb swelling, is caused by inactivating mutations in the VEGFR3 gene. Lymphedemadistichiasis syndrome, lymphedema associated with double rows of eyelashes, is caused by mutations in FOXC2. Hennecam syndrome, lymphedema with intestinal lymphangiectasia and intellectual disability, is caused by mutations in CCBE1, a gene that processes VEGF-C to its active form. Each syndrome reflects the specific role of the mutated gene in lymphatic development, and their study has illuminated the molecular roadmap of normal lymphangiogenesis. The concept of immune privilege, the state of certain anatomical sites including the eye, the brain, the testis, and the placenta, in which immune responses are actively suppressed to protect structures that cannot tolerate the collateral damage of inflammation, depends on multiple mechanisms including physical barriers, local immunosuppressive factors, and the induction of regulatory rather than effector immune responses. Immune privilege was first recognized in the context of organ transplantation, when it was noted that foreign tissue grafts, rejected rapidly when placed in most sites in the body, could survive for extended periods when placed in the anterior chamber of the eye. The eye was the first organ recognized as an immunologically privileged site, and the mechanisms responsible for this privilege have since been partially characterized. The blood-ocular barrier, a tight junctional barrier analogous to the blood-brain barrier, restricts the entry of immune cells and inflammatory mediators into the eye. The aqueous humor contains a cocktail of immunosuppressive factors including TGF-beta, alpha-melanocyte-stimulating hormone, and vasoactive intestinal peptide that suppress the activation of immune cells that do enter. The ocular surface cells express FosL, the ligand for Fos, that induces apoptosis in Fos-expressing T cells that attempt to infiltrate the tissues. And the immune cells that do enter the eye are preferentially skewed toward regulatory rather than effector function by the local environment. The brain employs similar mechanisms through the blood-brain barrier and the immunosuppressive environment of the CSF. The testis uses similar mechanisms to protect the developing sperm, which express novel antigens not present in the circulation until after immune education is complete, and which would therefore be recognized as foreign by an unregulated immune response, including a blood-testis barrier formed by Sertoli cell tight junctions and local TGF-beta production. Immune privilege is not absolute. Severe inflammatory stimuli can overcome these suppressive mechanisms, as in the autoimmune uveitis and encephalitis that can develop under severe inflammatory conditions. The measurement of lymph node involvement in cancer, the end staging of the TNM system, is one of the most powerful predictors of cancer prognosis and one of the key determinants of cancer treatment, reflecting the fact that the lymphatic vessels are the primary route of cancer cell dissemination from the primary tumor to distant sites. The pathological staging of cancer, the systematic assessment of the primary tumor, the regional lymph nodes, and the distant metastases, is the foundation of oncological decision-making, and the lymph node assessment is often the most prognostically significant component. Cancer cells that have invaded the tumor's stromal and vascular microenvironment can enter the lymphatic capillaries, which are more easily penetrated than blood capillaries, having larger pores and lacking a complete basement membrane, and travel in the lymph to the regional lymph nodes, where they may be trapped and eliminated by the immune response or may survive, proliferate, and form a lymph node metastasis. The presence or absence of lymph node metastases, and the number and location of involved nodes, is the primary determinant of stage in many cancers, including breast cancer, colorectal cancer, melanoma, and many others. A patient with N0 disease, no lymph node involvement, has an entirely different prognosis and requires different treatment than a patient with N2 or N3 disease. Extensive regional node involvement. The sentinel lymph node biopsy, the technique developed in the 1990s in which the first node to which a tumor drains is identified by injection of tracer around the tumor and selectively sampled, has transformed the staging of melanoma and breast cancer, allowing accurate nodal staging with minimal morbidity. If the sentinel node is free of tumor, the remaining regional nodes are very likely also free of tumor, and extensive lymph node dissection, with its associated risks of lymphedema, can be avoided. The tonsils and adenoids are characterized by their involution through adolescence and adulthood, reaching their maximum size relative to body size in childhood and progressively shrinking thereafter. a pattern that reflects the diminishing need for the first encounter immunological education that these strategically positioned sentinel organs provide as the adaptive immune repertoire matures. The tonsillar tissue size follows a predictable developmental trajectory that is one of the lymphatic system's most familiar clinical manifestations. In infancy and early childhood, the tonsils and adenoids are relatively small and immunologically inexperienced. Through middle childhood, approximately 4 to 7 years, they reach their peak size relative to the body, a period that coincides with the maximum first exposure rate to common respiratory pathogens and the most active phase of adaptive immune education. The tonsils and adenoids are enlarged during this period not only because they are actively processing the first exposures to many common organisms, but because they are sites of intense germinal center activity. The follicles are constantly undergoing the B-cell activation and affinity maturation that builds the immunological memory against the common respiratory viruses and bacteria. After approximately 7 to 10 years, the pace of first exposure slows. Most common organisms have been encountered and immunological memory established, and the tonsillar tissue begins its gradual involution. By early adulthood, the adenoid has usually involuted almost completely, and the palatine tonsils are much smaller than in childhood. This involution is normal and expected. The immune system no longer needs the sentinel structures at the maximum size required for first encounter education. The decision about tonsillectomy, the surgical removal of the palatine tonsils, is informed by this natural history. Chronic or recurrent infections in childhood, when the tonsils are at their most active and most hyperplastic, are the most common indication, and the decision must weigh the morbidity of the procedure against the impact of the infections. The process of class switching, in which a B cell changes the antibody class it produces from the initial IgM to IgG, IgA, or IgA by recombining the constant region genes, allows the same antigen binding specificity to be expressed in antibody classes with entirely different effector functions, optimizing the immune response for different types of threat. Every B cell begins its antigen-specific antibody production by making IgM, the large, pentameric antibody class that is expressed on the B cell's surface as the antigen receptor and is the first antibody secreted into the blood upon B cell activation. IgM is effective at complement activation and agglutination of pathogens, but it cannot cross the placenta, has a short serum half-life, and does not penetrate tissues effectively. The class switch recombination that occurs in the germinal center changes the constant region of the heavy chain that the B cell expresses, from the mu constant region that encodes IgM to the gamma constant regions that encode IgG, the alpha constant region that encodes IgA, or the epsilon constant region that encodes IgA, through a DNA recombination event that joins the variable region exons to the new constant region exons and deletes the intervening DNA. This recombination is irreversible. A B cell that has switched to IgG cannot go back to IgM, and is driven by the cytokine signals the B cell receives during the germinal center reaction. Interleukin-4 and interleukin-13 from Th2 cells drive switching to IgG1 and IgA. TGF-beta drives switching to IgA. Interferon gamma drives switching to IgG2A in mice and IgG1 in humans. The result is that the cytokine environment of the germinal center, which reflects the nature of the infection being responded to, determines which antibody class is produced. Intracellular pathogens that require cellular immunity generate Th1 cytokine environments favoring IgG subclasses. Parasitic infections and allergens generate Th2 cytokine environments favoring IgA. Mucosal pathogens generate conditions favoring IgA. The cisterna chyli, the dilated sac at the origin of the thoracic duct, located in the retroperitoneal space between the first and second lumbar vertebrae, receives the intestinal trunk and the two lumbar trunks that drain the lower extremities and the pelvic organs, and its presence in anatomy is relevant to surgeons operating in the vicinity of the abdominal aorta. The cisterna chyli is one of those anatomical structures that most people never encounter unless they have had occasion to look at cross-sectional imaging of the abdomen or to operate in the retroperitoneal space. It is a thin-walled, oval sac approximately 3 to 6 centimeters long, located just to the right of the abdominal aorta and just behind the right crus of the diaphragm, at approximately the level of the first and second lumbar vertebrae. It receives the intestinal trunk that carries the fat-laden chyle from the mesenteric lymphatics, and therefore becomes conspicuously white and distended after a fatty meal, and the left and right lumbar trunks that drain the lymph from the lower extremities and the pelvic and retroperitoneal organs. From the cisterna chyli, the thoracic duct descends through the aortic hiatus into the chest. The cisterna chyli is not always present as a discrete sac. In some individuals, it is replaced by a plexus of smaller vessels at the origin of the thoracic duct, and its anatomy is variable enough that its absence or altered configuration can create diagnostic confusion when searching for it on imaging. Its clinical significance arises primarily in the context of abdominal aortic surgery, spinal surgery, and retroperitoneal dissection, in which inadvertent injury to the cisterna chyli or the proximal thoracic duct can produce chylous ascites, the accumulation of milky, fat-laden lymph in the peritoneal cavity. Chylous ascites is a rare but serious complication that is diagnostically distinctive by the appearance and biochemistry of the acidic fluid and that may require dietary modification to reduce chylomicron production and thereby reduce flow through the damaged vessel, or surgical repair. The mechanism of lymphocyte recirculation, the continuous migration of lymphocytes from the blood into the lymph nodes through the high endothelial venules, transit through the lymph node parenchyma, exit via the efferent lymphatics, travel through the lymphatic vessels, and return to the blood via the thoracic duct, ensures that the rare antigen-specific lymphocyte can survey the entire body for its target antigen. The problem that lymphocyte recirculation solves is the problem of finding a needle in a haystack, or more precisely, of ensuring that a single lymphocyte specific for a particular antigen can encounter that antigen wherever it appears in the body. The immune system contains approximately 2 trillion lymphocytes, of which perhaps 1 in a million is specific for any given antigen. If these lymphocytes simply circulated in the blood, the rare antigen-specific cell would be unlikely to encounter its antigen, which is presented in the lymph nodes by dendritic cells that have migrated from the tissues. The recirculation pathway, in which lymphocytes continuously cycle from the blood through the lymph nodes and back to the blood, ensures that every lymphocyte visits every lymph node in the body approximately once every 12 to 24 hours. Each visit involves the lymphocyte entering the lymph node through the high endothelial venules, the specialized post-capillary venules whose cuboidal endothelial cells express the addressins that lymphocyte homing receptors recognize. Spending time in the appropriate zone of the lymph node sampling the presented antigens, and exiting via the efferent lymphatics if it has not found its cognate antigen. If a naive T cell encounters its antigen presented by a dendritic cell in the node, it stops migrating, becomes activated, and begins its program of proliferation and differentiation. The recirculation pathway is thus the lymphatic system's elegant solution to the probability problem of antigen encounter. By ensuring that every lymphocyte visits every lymph node regularly, it transforms a statistical improbability into a near certainty, given enough time. The lymphatic system's role in lipid-lowering therapy is highlighted by the observation that statins, which reduce hepatic cholesterol synthesis and increase LDL receptor expression, also have anti-inflammatory effects mediated partly through the modulation of lymphocyte and macrophage function, demonstrating the interplay between the metabolic and immune functions of the lymphatic system. Statins, the HMG-CoA reductase inhibitors that are among the most prescribed drugs in the world, were developed and are primarily used to reduce cardiovascular risk by lowering LDL cholesterol. But the clinical trials that demonstrated their cardiovascular benefit revealed reductions in cardiovascular events that exceeded what could be explained by cholesterol lowering alone, suggesting additional mechanisms. The pleiotropic effects of statins, their anti-inflammatory, immunomodulatory, and vascular effects independent of their cholesterol-lowering action, are now recognized as significant contributors to their clinical benefit, and many of these effects operate through the lymphatic immune system. Statins inhibit the isoprenoid pathway, reducing the availability of the lipid intermediates, geranyl pyrophosphate and farnesyl pyrophosphate, that are required for the prenylation of small GT possets including Rho, RAC, and RAS. These GT possets are essential for macrophage activation, lymphocyte proliferation, NK cell cytotoxicity, and the expression of adhesion molecules on the endothelium. By reducing prenylation, statins reduce the inflammatory activation of macrophages, the migration of monocytes into atherosclerotic plaques, the activation of T cells, and the expression of the endothelial adhesion molecules that capture circulating leukocytes. Statins also increased the expression of regulatory T cells in experimental models, suggesting a shift toward immunological tolerance that may contribute to their anti-inflammatory effects. The cardiovascular benefit of statins is thus, in part, a benefit mediated through the lymphatic immune system's role in the inflammatory biology of atherosclerosis. The high endothelial venule. The specialized post-capillary venule and lymph nodes and Peyer's patches through which lymphocytes enter the lymphoid organ from the blood, is characterized by its cuboidal endothelial cells that express tissue-specific addressins, and its biology is the foundation of the homing of specific lymphocyte subsets to specific lymphoid tissues. The high endothelial venule is the gateway through which the lymphocyte recirculation pathway enters the lymph node, the portal through which naive lymphocytes exit the blood and enter the lymphoid organ to survey for antigen. The venule gets its name from the unusual morphology of its endothelial cells, where the endothelium of ordinary post-capillary venules is flat and squamous. The endothelium of the high endothelial venule is tall and cuboidal, projecting into the lumen of the vessel. This tall morphology reflects the high metabolic activity of these specialized cells, which synthesize and present the tissue-specific addressins, the cell surface carbohydrate ligands that interact with lymphocyte homing receptors to initiate the tethering and rolling of lymphocytes on the venule surface. In the lymph nodes, the peripheral node addressin, PNAD, is expressed by the HEV endothelium and recognized by the lymphocyte homing receptor L-selectin, providing the initial tethering of naive lymphocytes. In the Peyer's patches and mesenteric lymph nodes, the mucosal addressin MADCAM1 is expressed and recognized by the lymphocyte receptor alpha-4 beta-7 integrin, the gut homing receptor expressed on lymphocytes that have been educated in the mucosal environment to return to the gut. This tissue-specific addressin expression is the molecular basis of lymphocyte homing. Different lymphocyte subsets express different homing receptors that match the addressins of different tissues, ensuring that gut-specific lymphocytes return to the gut, skin-specific lymphocytes return to the skin, and naive lymphocytes recirculate through the secondary lymphoid organs. The tumor microenvironment, the complex cellular and molecular milieu surrounding tumor cells in a solid tumor, is heavily infiltrated by immune cells including tumor-associated macrophages, regulatory T cells, and myeloid-derived suppressor cells that collectively establish an immunosuppressive environment that impairs the anti-tumor cytotoxic T cell response. The tumor microenvironment is, from the perspective of the immune system, a contested territory, a space in which the immune system's surveillance capacity is actively being suppressed by the tumor and the cellular allies it has co-opted. Among the cellular co-conspirators, the tumor-associated macrophages, typically polarized toward an M2-like, anti-inflammatory phenotype by the TGF-beta, IL-10, and IL-4 that tumors produce, are particularly important. These M2 macrophages produce the anti-inflammatory cytokines and growth factors that promote tumor growth and angiogenesis rather than the inflammatory cytokines and reactive oxygen species of the anti-tumor M1 macrophage. They degrade the extracellular matrix to facilitate tumor invasion and produce VEGF that promotes tumor angiogenesis. The regulatory T cells that accumulate in tumor microenvironments, attracted by tumor-produced CCL22 and CCL17, which activate CCR4 on TREGs, suppress the cytotoxic T cell responses that would otherwise recognize and kill tumor cells. The myeloid-derived suppressor cells, immature myeloid cells that expand dramatically in cancer-bearing hosts, produce arginase and inducible nitric oxide synthase, depleting the local arginine supply that T cells need for function and producing reactive nitrogen species that directly damage T cell signaling molecules. The result is a microenvironment in which potentially tumor-reactive T cells are surrounded but rendered ineffective. The immune surveillance that should eliminate the tumor neutralized by the tumor's own immunological self-defense. The phenomenon of leaky gut, the increase in intestinal epithelial permeability that allows bacterial products and larger antigenic molecules to pass more easily from the intestinal lumen into the lamina propria, is associated with systemic inflammation, and the lymphatic drainage of the increased antigenic load from the intestine may contribute to systemic inflammatory conditions including metabolic disease. The integrity of the intestinal epithelial barrier, maintained by the tight junctions between adjacent enterocytes that seal the paracellular space, is fundamental to the separation of the enormous microbial burden of the gut lumen from the sterile internal environment. When this barrier is compromised by infection, by dietary factors including high-fat diets, alcohol, and certain food additives, by psychological stress, or by dysbiosis of the microbiome, the paracellular permeability increases, allowing small amounts of lipopolysaccharide and other bacterial products, as well as larger food antigens, to enter the intestinal interstitium. The mucosal immune system normally contains this increased antigenic load through its dense network of lymphoid tissue, the lamina propria macrophages and dendritic cells sampling and processing the increased antigen, the IgA production of the lamina propria plasma cells neutralizing luminal bacteria. But when the permeability increase is substantial, some fraction of the bacterial products enters the mesenteric lymphatics and reaches the systemic circulation. Low-grade endotoxemia, the chronic presence of small amounts of bacterial lipopolysaccharide in the blood, produces the chronic low-grade systemic inflammation that is associated with obesity, insulin resistance, type 2 diabetes, and cardiovascular disease. The lymphatic drainage from the intestine, carrying not only chylomicrons but also the bacterial products and inflammatory mediators from the gut, is the route through which intestinal dysbiosis and barrier dysfunction communicate their metabolic consequences to the rest of the body. The lymphocyte's ability to traffic between the blood and the lymphoid organs is regulated by the sphingosine 1-phosphate signaling system, with lymphocytes exiting the lymph node when S1P in the lymph exceeds the intranodal S1P concentration, and the S1P receptor modulator fingolimod trapping lymphocytes in the lymph nodes as a therapeutic strategy for multiple sclerosis. Sphingosine 1-phosphate is a bioactive lipid mediator produced from sphingosine by the enzyme sphingosine kinase, and it is present in the blood and lymph at concentrations that are higher than in lymph node parenchyma. This concentration gradient, high S1P in the blood and lymph, low S1P in the lymph node, is the exit signal for mature lymphocytes that have completed their surveillance of the lymph node without encountering their cognate antigen. Lymphocytes express the S1P receptor S1PR1 on their surface, and when they sense the high S1P concentration at the lymph nodes' efferent lymphatic vessels, which are in continuity with the lymph, they migrate down the S1P gradient, exiting the lymph node through the efferent lymphatic and re-entering the recirculation pathway. Fingolimod, the first oral treatment approved for relapsing remitting multiple sclerosis, is a structural analog of sphingosine that is phosphorylated in the body to fingolimod phosphate, which binds to S1P receptors and causes their internalization and downregulation. Lymphocytes treated with fingolimod can no longer respond to the S1P gradient that normally drives their exit from lymph nodes, and they become sequestered in the lymph nodes, unable to recirculate, unable to traffic to the brain. The autoreactive T cells that are responsible for the inflammatory demyelination of multiple sclerosis cannot reach their target in the CNS, and the disease activity is reduced. The S1P receptor system is thus one of the most elegantly exploited therapeutic targets in the lymphatic immune system. The lymphatic system plays a critical role in the resolution of inflammation, not merely its initiation. Through the clearance of cellular debris, inflammatory mediators, and immune cells from inflamed tissues, and the impairment of this resolution phase by lymphatic dysfunction can lead to the chronic, unresolved inflammation that characterizes many autoimmune and inflammatory diseases. The resolution of inflammation is as actively regulated as its initiation, and the lymphatic system is central to both processes. During the acute inflammatory response, the lymphatic vessels of the inflamed tissue are exposed to a range of inflammatory mediators: histamine, prostaglandins, bradykinin, that increase their permeability and partially impair their contractile function, transiently reducing lymphatic drainage of the inflamed tissue. This initial impairment may serve the purpose of concentrating immune cells in the inflamed tissue during the early, high-activity phase of the response. As the inflammatory response begins to resolve, as the pathogens are cleared, as the apoptotic neutrophils are phagocytosed by macrophages, as the anti-inflammatory mediators including lipoxins, resolvins, and protectins accumulate, the lymphatic vessels are stimulated to increase their drainage rate. The resolving macrophages release VEGF-C, stimulating lymphangiogenesis and increasing the functional capacity of the lymphatic drainage. The increased lymphatic flow clears the debris, inflammatory mediators, and spent immune cells from the tissue, reducing the inflammatory burden and creating the conditions for tissue repair. In chronic inflammatory diseases, rheumatoid arthritis, inflammatory bowel disease, psoriasis, the lymphatic vessels in the affected tissues may be structurally abnormal or functionally impaired, contributing to the failure of resolution. The lymphatic vessels of chronically inflamed joints in arthritis models show dilated, dysfunctional vessels with impaired contractility, a finding that may contribute to the chronicity of joint inflammation by preventing the clearance of the mediators that sustain it. There are so many places left to go in the lymphatic system tonight. The biology of allergic responses, the extraordinary world of the tumor-fighting T cell, the mucosal immunity that protects the gut, the molecular choreography of lymphocyte trafficking. If you would like to be here for those journeys, and for all the other quiet wonders that relax with science brings to the night, one slow evening at a time, subscribing keeps the door open between us. Something will be waiting on an evening when you need it. Made slowly. Made carefully. Made for exactly this hour. The IgA antibody class, produced in response to parasitic infections and allergens, characterized by its binding to high-affinity IgA receptors on the surface of mast cells and basophils, is the mediator of immediate hypersensitivity reactions including allergic rhinitis, asthma, urticaria, and anaphylaxis, and the excessive IgA responses of atopic individuals represent a dysregulated immune response to otherwise harmless antigens. IgA is the rarest antibody class in the circulation, present at concentrations of nanograms per milliliter compared to the milligrams per milliliter of IgG, but its functional impact is wildly disproportionate to its abundance. This disparity reflects IgA's unique mechanism of action, rather than neutralizing antigens or activating complement in the fluid phase as IgG does. IgA binds through its Fc region to the high-affinity IgA receptor Fc on the surface of mast cells and basophils, sensitizing these cells for future antigen encounters. A single IgE-sensitized mast cell can carry approximately 500,000 IgA molecules on its surface, each waiting for its specific antigen. When the antigen subsequently arrives, a pollen grains allergen, a bee venom protein, a food protein, it cross-links two or more of the surface-bound IgA molecules by binding simultaneously to both, triggering the rapid, massive degranulation of the mast cell, the explosive release of histamine, prostaglandins, leukotrienes, and proteases that produces the symptoms of the immediate allergic reaction, the sneezing and congestion of rhinitis, the bronchoconstriction of asthma, the urticaria and angioedema of skin allergic responses, the cardiovascular collapse and airway edema of anaphylaxis. In evolutionary terms, this IgE mast cell system is thought to have evolved as a defense against multicellular parasites, helminth worms, which trigger IgA responses and which are defended against by the tissue damage and smooth muscle contraction that mast cell degranulation produces. The atopic individual's IgA response to environmental allergens is a misdirected version of this anti-parasite defense. The process of antigen processing and presentation, the pathway by which a cell takes up antigen, degrades it into peptides, and loads the resulting peptides onto MHC molecules for display to T cells, is fundamentally different for MHC class I and MHC class II, reflecting the different surveillance needs of the cytotoxic and helper T cell arms of immunity. MHC class I and MHC class II molecules present peptides to different T cell populations for different purposes, and the pathways by which peptides are generated and loaded onto these molecules are correspondingly different. MHC class I, expressed on virtually every nucleated cell in the body, presents peptides from proteins synthesized within the cell. The proteasome, the cytoplasmic protein degradation complex, continuously degrades intracellular proteins into short peptides of 8 to 10 amino acids. These peptides are transported into the endoplasmic reticulum by the TAP transporters, where they are loaded onto newly synthesized MHC class I molecules with the assistance of chaperone proteins. The MHC class I peptide complex travels through the Golgi to the cell surface, where it can be surveyed by CD8 cytotoxic T cells. Any peptide derived from an intracellular protein, viral protein, tumor antigen, self-protein, can be presented on MHC class I, making every nucleated cell a potential target for cytotoxic T cell killing if it is producing a foreign or altered protein. MHC class II, expressed primarily on professional antigen-presenting cells, dendritic cells, macrophages, and B cells, presents peptides from proteins taken up by endocytosis. The engulfed proteins are degraded in the endosomal lysosomal pathway into peptides of 13 to 25 amino acids, which are loaded onto MHC class II molecules in a specialized compartment, the MIIC, and transported to the cell surface. This class II pathway presents what the cell has been eating: extracellular proteins, bacterial antigens, immune complexes, to CD4 T helper cells, providing information about what is present in the extracellular environment. The lymphatic system's involvement in tumor metastasis. Through the lymphatic invasion by tumor cells, their transport to regional lymph nodes, and their potential establishment of lymph node micrometastases, has made the lymphatic vessels and lymph nodes central to cancer staging, treatment planning, and the development of anti-metastatic therapeutic strategies. Cancer metastasis, the spread of cancer cells from the primary tumor to distant sites, where they establish secondary tumors, is responsible for the majority of cancer deaths, and the lymphatic route is, for most carcinomas, the initial pathway of dissemination. The process begins when tumor cells at the invasive front of the primary tumor breach the extracellular matrix of the tumor stroma and enter the lymphatic capillaries, which are relatively easier to penetrate than blood capillaries due to their lack of continuous basement membrane and their button junction organization. Once in the lymphatic lumen, tumor cells travel in the lymph to the regional lymph nodes, where they may be trapped and killed by the immune response or may survive and establish a lymph node metastasis. The biology of this survival in the lymph node is complex. The lymph node environment includes both tumoricidal components, NK cells, cytotoxic T cells, activated macrophages, and tumor-supportive components, the follicular dendritic cell network, the regulatory T cell population, the anti-inflammatory cytokine milieu of the normal lymph node. Whether tumor cells survive in the lymph node and go on to see distant metastases depends on the balance of these factors and on the specific molecular properties of the tumor cells. The development of the sentinel lymph node technique has allowed the minimal morbidity assessment of nodal involvement without extensive lymph node dissection, and the genomic analysis of lymph node micrometastases has revealed information about the molecular characteristics of the metastatic cell subpopulation that may guide treatment decisions. The lymphatic system undergoes significant changes during aging, including the involution of the thymus, the reduction in naive T-cell production, the accumulation of memory and effector cells at the expense of naive cell diversity, and the decline in lymph node germinal center activity, collectively termed immunosenescence, which contributes to the increased infection susceptibility and reduced vaccine responsiveness of elderly individuals. Immunosenescence, the progressive deterioration of immune function with advancing age, is one of the most consequential biological changes of aging, contributing to the increased susceptibility to infectious disease, the reduced response to vaccination, the increased risk of cancer, and possibly to the chronic low-grade inflammation sometimes called inflammaging that is associated with many age-related diseases. The thymic component of immunosenescence is the most fundamental, as the thymus involutes through adulthood, progressively replaced by fat until relatively little functional thymic parenchyma remains by the 6th or 7th decade. The production of new naive T cells from thymic immigration falls. The peripheral naive T cell pool is maintained in younger adults primarily by thymic output and by homeostatic proliferation of existing naive T cells. With age, the thymic contribution diminishes and homeostatic proliferation becomes the primary maintenance mechanism. The proliferating naive T cells acquire some features of memory cells: reduced expression of CD28, increased expression of CD57, and their diversity progressively narrows as certain clones expand at the expense of others. The B cell compartment is similarly affected. The germinal center reactions of elderly individuals are smaller, produce lower affinity antibodies with less class switching, and generate fewer long-lived plasma cells and memory B cells than those of younger individuals. These changes underlie the reduced immunogenicity of many vaccines in elderly people, and the need for higher dose or adjuvant vaccine formulations in this population. The blood-brain barrier's unique tight junction endothelium does not have a conventional lymphatic drainage counterpart in the brain parenchyma, but the recently characterized meningeal lymphatic vessels, running along the dural sinuses and foramina, provide a drainage pathway for brain interstitial fluid and cerebrospinal fluid that connects the central nervous system to the cervical lymphatic system. The discovery of meningeal lymphatic vessels, first clearly characterized by Jonathan Kipnis and colleagues in 2015, building on earlier work by several groups, resolved the long-standing puzzle in neuroscience. How does the brain drain its interstitial fluid and cerebrospinal fluid in the absence of conventional lymphatic vessels? The meningeal lymphatic vessels run along the dural venous sinuses and through the foramina at the skull base, particularly the cribriform plate in the jugular foramen, and drain CSF that has been delivered to the meningeal spaces from the brain parenchyma via the glymphatic system. From the meningeal lymphatics, the fluid drains to the deep cervical lymph nodes and thence into the cervical lymphatic system and the thoracic duct. The meningeal lymphatics are thin-walled vessels with the molecular signature of lymphatic endothelium, expressing LYVE1, podoplanin, and VEGFR3, and they respond to VEGF-C with lymphangiogenesis, demonstrating that they are functional lymphatic vessels in the conventional sense. Their function in normal physiology appears to include the drainage of CSF and the transport of immune cells and antigens from the meningeal spaces to the cervical lymph nodes, providing a surveillance pathway through which the central nervous system's immune state can be communicated to the peripheral adaptive immune system. Their dysfunction with aging. The meningeal lymphatics deteriorate in efficiency in older mice, coinciding with impaired lymphatic clearance, may contribute to the age-related accumulation of amyloid beta and other proteinaceous debris in the aging brain. The lymphatic system's role in fat absorption makes it a route of administration for certain drugs, particularly lipophilic molecules that, when formulated with lipid excipients and taken orally, can be absorbed with chylomicrons via the lacteals and bypass the hepatic first-pass metabolism that limits the bioavailability of many drugs taken by the conventional portal venous route. The pharmacological exploitation of the intestinal lymphatic route, giving drugs via the lymphatics rather than the portal blood, is a strategy with theoretical and, increasingly, practical appeal in drug delivery. The conventional absorption of orally administered drugs involves passage through the intestinal epithelium into the portal blood, travel through the portal vein to the liver, and exposure to hepatic metabolism before the drug reaches the systemic circulation. For drugs with high hepatic extraction ratios, high first-pass metabolism, this hepatic exposure dramatically reduces bioavailability. A drug that is 90% extracted by the liver on its first pass reaches the systemic circulation at only 10% of the absorbed dose. The intestinal lymphatic route bypasses the liver entirely. Drugs that can be formulated to associate with chylomicrons, typically lipophilic drugs, with log p-values above 5, that can incorporate into the lipid core or surface of the lipoprotein particle, are absorbed with the chylomicrons via the lacteals, travel through the mesenteric lymphatics and the thoracic duct, and enter the venous blood at the subclavian vein without passing through the liver. For drugs with high hepatic extraction, this bypass can dramatically increase bioavailability. The anti-HIV drug lopinavir and the anti-cancer agent paclitaxel have both been formulated with lipid excipients to enhance lymphatic absorption. The challenge is that the lymphatic route depends on chylomicron formation, which requires a fat-containing meal, making the efficacy of lymphatically absorbed drugs food-dependent in a way that standard oral formulations are not. The lymphocyte's extraordinary capacity for self-renewal. The homeostatic proliferation of both naive and memory lymphocytes in response to survival signals from cytokines including interleukin-7 and interleukin-15 and from weak T-cell receptor signaling by self-MHC, maintains the lymphocyte pool at a relatively stable size throughout adult life despite the progressive decline in thymic output. The maintenance of the peripheral lymphocyte pool at approximately 2 trillion cells throughout adult life is achieved not by a constant rate of thymic export, which declines progressively through adulthood, but by the homeostatic proliferation of the existing peripheral lymphocytes. Homeostatic proliferation is driven by two categories of signal: cytokine survival signals and tonic T-cell receptor signaling from self-MHC molecules. The key cytokines are interleukin-7, produced by stromal cells of the lymph nodes in bone marrow and essential for both naive T cell and B cell survival, and interleukin-15, produced by dendritic cells and macrophages and essential for memory CD8 T cell and NK cell survival. When the lymphocyte pool is depleted by lymphopenia from chemotherapy, from HIV infection, or from experimental manipulations in animal models, the surviving lymphocytes proliferate more rapidly, driven by the increased availability of IL-7 and IL-15 that the reduced pool releases from competition. This lymphopenia-induced proliferation is the mechanism by which T cell reconstitution occurs after bone marrow transplantation and after immune ablation therapy. The weak TCR signals from self-MHC that contribute to homeostatic proliferation of naive T cells also introduce a risk. Some self-reactive T cells that were anergic in a replete immune system may escape anergy during lymphopenic proliferation, when the signals for survival override those for tolerance, a mechanism that may contribute to the autoimmune phenomena sometimes observed after lymphocyte depleting therapy. I wonder how you are feeling right now. Whether the body has found some of the ease it was looking for when it arrived here tonight. Whether the lymph is moving through its quiet channels without your attention, as it always does, as it has always done. Whether the breath is a little slower. Whether the thoughts have softened from whatever they were an hour ago. If any of that is true, even a fraction, I am glad. And if you feel like leaving something in the comments about what tonight feels like for you, I would find that genuinely meaningful. Just, I was here. The system was at work. The quiet was real. That is enough. The lymphatic system's development continues postnatally, with the colonization of the gut by the microbiome driving the maturation of the mucosal immune system and the development of the gut-associated lymphoid tissue, and the composition of the early microbiome, shaped by mode of delivery, feeding, and antibiotic exposure, as lasting effects on immune development. The gut microbiome and the mucosal lymphatic immune system develop in a reciprocal dialogue during the first years of life that shapes the immune system's long-term character. At birth, the gut is essentially sterile, and the gut-associated lymphoid tissue, the Peyer's patches, the isolated lymphoid follicles, the lamina propria lymphocytes, is relatively undeveloped compared to its adult form. The colonization of the gut by bacteria in the first days and weeks of life drives the maturation of these structures. The microbial antigens and microbial metabolites stimulate the development of the Peyer's patches, the expansion of the lamina propria lymphocyte populations, and the establishment of the regulatory T cell pool that maintains tolerance to the microbiome. Germ-free animals, raised in sterile conditions without any microbial exposure, have dramatically underdeveloped gut-associated lymphoid tissue, with small Peyer's patches containing few germinal centers and reduced lamina propria IgA-producing plasma cells. Recolonization of germ-free animals with defined microbial communities restores normal lymphoid development, confirming that the microbiome is the driving signal. In human infants, the composition of the colonizing microbiome, shaped by whether the infant is delivered vaginally or by cesarean section, whether it is breastfed or formula-fed, whether it receives antibiotics, has lasting effects on the character of the immune system that develops. Infants colonized by a diverse, bacterial-rich microbiome develop more robust regulatory T-cell populations, higher secretory IgA production, and lower rates of allergic disease in later childhood, consistent with the hygiene hypothesis that reduced early microbial exposure contributes to the rising prevalence of atopic disease in high-income countries. The phenomenon of bystander activation, the activation of lymphocytes that are not specific for the antigen that initiated the immune response, through the inflammatory cytokines produced by the ongoing response, may contribute to the exacerbations of autoimmune diseases that occur during viral infections, as the inflammatory environment nonspecifically activates pre-existing autoreactive T cells. The adaptive immune response is generally described as antigen-specific. Only the lymphocytes whose receptors match the antigen are activated. This is mostly true, but the inflammatory environment created by an immune response can also nonspecifically activate lymphocytes in the vicinity, a phenomenon called bystander activation. During a vigorous antiviral immune response, the high concentrations of inflammatory cytokines, interleukin-2, interleukin-15, interferon gamma, create a mitogenic environment that can drive the proliferation of memory T cells in the vicinity, even if those memory T cells are specific for a completely different antigen. Similarly, the activation of dendritic cells and macrophages during infection can upregulate their expression of co-stimulatory molecules and increase their presentation of self-peptides on MHC class II, potentially providing the second and third signals that would activate autoreactive T cells that were previously held in anergy by the absence of co-stimulation. The clinical relevance of bystander activation to autoimmune disease is suggested by the well-known triggering of autoimmune flares by viral infections. Patients with multiple sclerosis, rheumatoid arthritis, and systemic lupus erythematosus frequently experience disease exacerbations in the context of respiratory infections. Whether this reflects bystander activation of autoreactive lymphocytes, molecular mimicry between viral and self-antigens, or indirect effects of the inflammatory cytokines on the target tissues is an area of active investigation, but bystander activation provides a mechanistic framework for understanding how a non-specifically triggered immune response can have consequences for self-reactive immune populations. The lymphatic microvalves, the intraluminal valves of the precollector and collector lymphatic vessels, were first described in detail by 1787 by Paolo Mascagni, whose meticulous anatomical illustrations of the lymphatic system remained a standard reference for over a century and constituted one of the first systematic mappings of the lymphatic vasculature. The history of the lymphatic system's discovery is somewhat less celebrated than the histories of other organ systems. There is no Harvey for the lymphatics in the way that William Harvey's 1628 description of the circulation transformed cardiovascular biology. The discovery of the lymphatic system was piecemeal and contested. Gasparo Uccelli first described the milky lacteal vessels of the small intestine in 1627, while dissecting a well-fed dog. Thomas Bartholin, Jean Piquet, and Olaf Rudbeck each contributed to the mapping of the thoracic duct and the discovery that the lymphatic system drained into the venous blood rather than into the liver, as had been assumed, through work in the mid-17th century. But it was Paolo Mascagni, an Italian anatomist working in the late 18th century, who produced the first comprehensive mapping of the human lymphatic system, based on the meticulous injection of mercury into lymphatic vessels, the technique of choice for visualizing the system's fine detail before any imaging technology existed, and careful drawing of the results. Mascagni's 1787 work Vasorum Lymphaticorum Corporis Humani Historia et Ichnographia contained illustrations of such detail and accuracy that they remained a standard anatomical reference for over a century, and his characterization of the lymphatic valves, the bicuspid intraluminal valves that prevent lymph backflow, provided the structural basis for understanding directional lymph flow. The lymphatic system's relative neglect in the subsequent history of biomedical science, compared to the cardiovascular and nervous systems, reflects in part the difficulty of studying a system whose vessels are small, invisible, and empty of the colored fluid that makes the blood vascular system easy to trace. The lymphatic system's role in the immune response to vaccines, carrying the vaccine antigen to the regional lymph nodes via the afferent lymphatics, and transporting the antigen-presenting dendritic cells that initiate the T and B cell responses, means that the site of vaccine injection and the local lymphatic anatomy determine the efficiency of the immune response to vaccination. The site of vaccine injection determines which regional lymph nodes receive the vaccine antigen and which lymphocyte populations are initially exposed to it, and these anatomical factors can influence both the magnitude and the quality of the immune response. For most intramuscular vaccines, injected into the deltoid muscle of the upper arm or the vastus lateralis of the thigh, the antigen drains through the muscle's lymphatic capillaries into the regional collecting vessels and reaches the axillary or inguinal lymph nodes, respectively, within hours. The dendritic cells that have taken up vaccine antigen at the injection site migrate via the lymphatic vessels to the same regional nodes, arriving over 12 to 24 hours and initiating the T and B cell responses. The local inflammation at the injection site, produced by the adjuvant components of most vaccines, stimulates local lymphatic flow, increases dendritic cell maturation and migration, and upregulates the co-stimulatory molecules and cytokines that the dendritic cells need to activate naive lymphocytes in the node. Intradermal vaccine administration, injection into the dermis rather than the muscle, reaches the dense network of cutaneous lymphatic capillaries more directly and may produce a more efficient antigen delivery to the regional nodes with smaller antigen doses, an approach used in some influenza and rabies vaccine protocols where the antigen is scarce or expensive. The adjuvant formulations of modern vaccines are designed, in part, to optimize the lymphatic delivery of antigen by creating a depot effect at the injection site, slowing the antigen's entry into the lymphatics, providing a prolonged signal to the draining lymph nodes. The phenomenon of tolerance induction, the active establishment of non-responsiveness to specific antigens, can be achieved through the lymphatic route, and the tolerance to dietary antigens into the gut microbiome depends on the specific cytokine environment and cell populations of the mesenteric lymph nodes through which food antigens and microbial products are first processed. The mesenteric lymph nodes, the large cluster of lymph nodes draining the gastrointestinal tract, numbering approximately 100 to 150 in the adult human, occupy a unique position in the immune system. They are the site at which the immune system makes its most consequential decisions about tolerance. Food antigens, commensal microbial antigens, and self-antigens of the gut epithelium all arrive in the mesenteric nodes via the mesenteric lymphatics, and the immune response to these antigens must, under normal conditions, be one of tolerance. The induction of regulatory T cells and IgA-secreting B cells rather than the Th1, Th2, or Th17 effector responses that would produce inflammatory bowel disease, food allergy, or autoimmunity. The mechanisms by which the mesenteric lymph nodes induce tolerance rather than immunity to these normally harmless antigens include the specialized character of the dendritic cells that present antigen in this environment. The mesenteric node dendritic cells are conditioned by retinoic acid from the gut epithelium to preferentially induce FOXP3+ regulatory T cells rather than effector T cells, and the production of TGF-beta and interleukin-10 by the abundant regulatory T cell population of the mesenteric nodes. The failure of these tolerance mechanisms, producing the inappropriate immune responses to dietary gluten and celiac disease, to commensal bacteria and inflammatory bowel disease, or to the gut's own antigens and the autoimmunity of a poorly regulated immune system, is one of the most clinically consequential examples of lymphatic immune system failure. The concept of the immune synapse, the specialized junction that forms between a T cell and its antigen-presenting cell upon T cell receptor recognition, in which the signaling molecules are organized into a central supramolecular activation cluster surrounded by a ring of adhesion molecules, has illuminated the molecular basis of T cell activation and the mechanisms by which T cells integrate activating and inhibitory signals. The immune synapse was first described by Michael Dustin and colleagues in 1998, using fluorescence microscopy to visualize the interface between a T cell and an antigen-presenting cell. Rather than a diffuse, unorganized interface, they found a highly organized structure in which the T cell receptor and its associated signaling molecules, the CD3 complex, ZAP70, and downstream kinases, concentrated in the center of the interface, forming what they called the central supramolecular activation cluster, surrounded by a peripheral ring of the integrin LFA1 and its ligand ICAM1, which provides the adhesion that holds the two cells together. This organized junction, which forms over approximately 10 minutes of cell-cell contact and involves the active reorganization of the T cell's cytoskeleton, is the physical basis of efficient T cell activation, concentrating the signaling machinery at the junction where the TCR-PMHC interaction occurs and allowing the sustained, focused signaling that is required for full T cell activation. The immune synapse also organizes the inhibitory signals, the inhibitory receptor CTLA4, the target of the checkpoint inhibitor ipilimumab, is recruited to the immune synapse alongside CD28, where it competes with CD28 for binding to B7 on the APC surface. PD1, the target of nivolumab and pembrolizumab, similarly localizes to the immune synapse, where its engagement with PDL1 on the APC or tumor cell delivers the inhibitory signal that terminates T-cell activation. The immune synapse is the molecular stage on which the balance between activation and inhibition is decided. The lymphatic system's connection to the gut microbiome extends beyond the mesenteric lymphatics to include the trafficking of bacteria-derived extracellular vesicles, small membrane-enclosed particles carrying bacterial antigens, nucleic acids, and metabolites, through the lymphatic system to distant organs, potentially explaining some of the systemic effects of microbiome composition on non-gut physiology. The gut microbiome's influence on remote organ physiology, the brain, the liver, the immune system, the cardiovascular system, has been one of the most fertile areas of research in biomedical science of the past decade. And the lymphatic system provides one of the pathways through which this influence is mediated. Intestinal bacteria continuously shed small, membrane-enclosed extracellular vesicles, nanoscale particles of approximately 20 to 300 nanometers, that carry a cargo of bacterial antigens, lipopolysaccharide, small RNAs, metabolites, and other bioactive molecules. These vesicles can penetrate the mucus layer of the intestine, cross the epithelial barrier, and enter the lamina propria, from which they gain access to the lymphatics and travel to the mesenteric lymph nodes. Some fraction of bacterial extracellular vesicles also enters the blood, either directly or via the lymphovenous connection at the thoracic duct drainage. The systemic distribution of these vesicles provides a mechanism by which microbiome-derived signals can reach and influence remote tissues, the liver, through the portal blood, the brain, through vesicles that may penetrate the blood-brain barrier under inflammatory conditions, the cardiovascular system, through endothelial effects. The composition of bacterial extracellular vesicles reflects the composition of the producing microbiome, providing a dynamic readout of microbiome status that is continuously delivered to the host immune system. The lymphatic trafficking of these vesicles is therefore one of the mechanisms through which the microbiome-host dialogue is conducted, a molecular postal service whose envelopes carry microbial dispatches to every corner of the body. The regulatory role of the lymphatic endothelium in immune responses, through the expression of adhesion molecules, the production of chemokines including CCL21, and the induction of tolerance in T cells that encounter antigen on the lymphatic endothelial surface, positions the lymphatic vessel not merely as a conduit but as an active participant in immune regulation. The lymphatic endothelium's immunological role has expanded considerably in the scientific understanding developed over the past 2 decades, from a simple conduit for the passage of immune cells to an active participant in the regulation of immune responses. The observation that lymphatic endothelial cells express the chemokine CCL21, which attracts CCR7 expressing dendritic cells and naive T cells, established that the lymphatic endothelium actively directs the migration of immune cells toward the lymphatic vessel. But more surprising was the discovery that lymphatic endothelial cells also express MHC class II and can present antigens directly to CD4 T cells, and that the presentation of antigen by lymphatic endothelial cells, in the absence of the co-stimulatory molecules that professional antigen-presenting cells express, can induce energy rather than activation. Lymphatic endothelial cells in the lymph node sinuses present peripheral tissue antigens that they have acquired from the lymph, creating a central tolerance-like mechanism in the peripheral tissues, where self-antigens carried by the lymph from the skin and other tissues are presented to peripheral T cells under energizing conditions. This endothelial antigen presentation may contribute to the maintenance of peripheral tolerance to tissue-specific antigens. The lymphatic endothelium also produces the scavenging receptor LYVE1, which binds hyaluronan, the glycosaminoglycan that accumulates in the extracellular matrix of inflamed tissues, providing a mechanism for the lymphatic capillaries to clear hyaluronan from the inflamed interstitium and potentially modulate the inflammatory response through its removal. The phenomenon of tumor-induced lymphangiogenesis, the production by tumors of VEGF-C that stimulates the growth of new lymphatic vessels within and around the tumor, correlates with lymph node metastasis and reduced survival in many cancers, and the blocking of VEGF-C signaling has been investigated as an anti-metastatic therapeutic strategy. The relationship between VEGF-C production by tumors and lymph node metastasis is one of the more mechanistically clear connections in tumor biology. Tumors that produce high levels of VEGF-C, through the upregulation of the VEGF-C gene by hypoxia, inflammatory signals, and oncogenic signaling pathways, stimulate the growth of new lymphatic vessels within the tumor stroma and in the peritumoral tissue through the VEGFR3-dependent lymphangiogenesis pathway. These new lymphatic vessels are structurally abnormal compared to normal lymphatics. They may be dilated, poorly valved, and with irregular junctions, but they are functional to the extent that they can carry tumor cells that have entered their lumens. In animal models, blocking VEGF-C signaling, using neutralizing antibodies against VEGF-C or soluble decoy receptors that bind VEGF-C and prevent it from reaching VEGFR3 on lymphatic endothelial cells, reduces the density of intratumoral and peritumoral lymphatics and reduces the frequency of lymph node metastasis. In human cancers, VEGF-C expression by the primary tumor correlates with lymphatic vessel density, lymph node involvement, and reduced disease-specific survival in breast cancer, colorectal cancer, gastric cancer, and many others. These observations have motivated the development of anti-VEGF-C strategies as components of anti-metastatic therapy, either as standalone treatments or as adjuncts to conventional cancer therapy, although the translation from animal models to clinical benefit in humans has proven, as with many promising anti-cancer strategies, considerably more challenging than the preclinical results suggested. The complement system's role in lymphocyte activation, through the opsonization of antigen by C3d, which binds to the complement receptor CR2 on B cells and dramatically lowers the antigen threshold for B cell activation, explains why complement-coded antigens are orders of magnitude more immunogenic than uncoded antigens, and why complement deficiency impairs antibody responses. The discovery that complement opsonization dramatically enhances B cell responses to antigen by a factor of approximately 1,000 to 10,000 in some experimental systems, revealed an unexpected mechanism by which the innate immune system amplifies the adaptive immune response. The mechanism involves the complement fragment C3d, which is covalently attached to antigen-antibody complexes and to pathogen surfaces during complement activation. C3d serves as a danger signal, a molecular tag indicating that the immune system has already identified this antigen as requiring a response. B cells express the complement receptor CR2, CD21, which binds to C3d. When a B cell's surface immunoglobulin binds to an antigen that also carries C3d, the immunoglobulin antigen C3d complex simultaneously engages both the B cell receptor and CR2, creating a molecular bridge that dramatically amplifies the intracellular signaling from the B cell receptor. The co-receptor effect. This co-receptor signal reduces the threshold of antigen concentration required to activate the B cell by approximately 1,000-fold, explaining why complement-coded antigens are so much more immunogenic. In patients with complement deficiencies, particularly deficiencies of C3 or of the components that deposit C3d on antigens, antibody responses to complement-activating antigens are markedly impaired, explaining the susceptibility to recurrent bacterial infections that characterizes these patients. The complement B cell co-receptor system is also the mechanism by which the hepatitis B surface antigen vaccine and similar subunit vaccines generate strong antibody responses despite containing only a small amount of antigen. The adjuvants in these vaccines activate complement, generating the C3d opsonization that amplifies the B cell response. The lymph nodes sinusoidal macrophages, occupying the subcapsular sinus, the cortical sinuses, and the medullary sinuses, perform essential filtration functions capturing blood-borne pathogens and particulate antigens from the lymph, degrading cellular debris, and relaying captured antigens to the underlying lymphocytes. The sinus system of the lymph node, the channels through which the lymph percolates as it travels from the afferent lymphatics to the efferent lymphatic, is lined by specialized macrophages whose function is to capture and process the particulate and microbial content of the incoming lymph. The subcapsular sinus macrophages, expressing the mannose receptor, the scavenger receptors, and specific markers like SINR1, are immediately beneath the capsule, in the first region of the node that the afferent lymph enters. These cells are notable for their ability to rapidly capture and hold large particulate antigens, viral particles, bacteria, immune complexes, from the lymph and relay them intact to the underlying B cells in the follicles, providing the B cells with native antigen in a form that allows them to process and present it, or to be directly stimulated by it through their surface immunoglobulin. The rapid relay of native antigen from the subcapsular sinus macrophage to the follicular B cell can occur within hours of antigen arrival in the node, initiating the B cell response long before the conventional dendritic cell T cell B cell collaboration pathway would generate a B cell response. The medullary sinus macrophages, at the efferent end of the node, perform a final filtration function, capturing any antigens or organisms that have passed through the cortical regions of the node and providing a last line of defense before the filtered lymph exits via the efferent lymphatic. The role of the lymphatic system in drug delivery and pharmacokinetics has gained increasing attention with the recognition that drugs that distribute into the interstitial fluid and drain via the lymphatics may have different pharmacokinetic profiles, including prolonged tissue residence times and different routes of elimination, than drugs that distribute entirely through the blood. The traditional pharmacokinetic analysis of drug distribution assumes that drugs distribute between the blood and the tissues and are eliminated primarily through hepatic metabolism and renal excretion. For drugs that are sufficiently large or hydrophilic to distribute significantly into the interstitial fluid, however, the lymphatic drainage of the interstitium provides an additional route of distribution and return to the circulation, one that operates on a different timescale than the rapid equilibration between blood and well-perfused tissues that pharmacokinetic models typically assume. Monoclonal antibodies, large proteins of approximately 150 kilodaltons, are particularly likely to distribute into the interstitium and drain via the lymphatics, because their size prevents them from crossing capillary walls into the blood by simple diffusion, and they must instead move through the tissue interstitium and enter the lymphatic system before returning to the venous blood via the thoracic duct. Subcutaneously injected monoclonal antibodies are deliberately formulated for lymphatic absorption. The antibody is deposited in the subcutaneous interstitial space, distributes through the subcutaneous tissue, and enters the regional lymphatic capillaries, which carry it to the regional lymph nodes and then to the thoracic duct. This lymphatic absorption produces a slower, more sustained rise in plasma concentration than direct intravenous injection, potentially reducing the peak concentration-related adverse effects of these powerful drugs while maintaining therapeutic concentrations for extended periods. The subcutaneous formulations of many monoclonal antibodies, including adalimumab, rituximab, and trastuzumab, exploit this lymphatic pharmacokinetics for patient convenience and improved tolerability. The lymphatic system's contribution to the immune response to cancer immunotherapy, particularly to the checkpoint inhibitor drugs that unleash the immune system's anti-tumor activity, depends on the integrity of the draining lymph nodes, where the reinvigoration of exhausted tumor-specific T cells must occur before they can travel back to the tumor to exert their anti-tumor effects. The success of checkpoint inhibitor immunotherapy, which has produced remarkable long-term responses in many cancers including melanoma, lung cancer, and kidney cancer, depends on the ability of the immune system to generate and maintain the anti-tumor T cell responses that the checkpoint blockade has unleashed. The lymph node is central to this process. Before anti-PD-1 therapy can produce clinical benefit, The tumor-draining lymph nodes must contain a population of tumor antigen-specific T cells, either naive T cells that have been primed by dendritic cells carrying tumor antigens from the primary tumor, or exhausted effector T cells that have been rendered hyporesponsive by chronic PD-1 signaling in the tumor microenvironment. Anti-PD-1 therapy can reinvigorate these exhausted T cells both in the tumor microenvironment and in the tumor-draining lymph nodes, where the PD-1-PDL1 interaction between exhausted T cells and dendritic cells expressing PDL1 normally sustains the exhausted state. In the lymph node, the reinvigorated T cells receive the proliferative and survival signals they need to expand to large numbers before re-entering the circulation and trafficking back to the tumor. Surgical removal of the tumor-draining lymph nodes, which may be performed to stage the cancer or to remove lymph node metastases, impairs the ability of checkpoint inhibitor therapy to generate systemic anti-tumor responses, because the lymph node population of tumor-specific T cells that the therapy needs to reinvigorate has been removed. This finding has influenced the sequence of checkpoint inhibitor therapy in surgery in some cancer treatment protocols. The lymphocyte's journey from the bone marrow or thymus to its tissue homing location is guided by a series of homing receptor and addressin interactions, collectively the multi-step adhesion cascade, that is remarkably tissue-specific, ensuring that lymphocytes educated in the gut return to the gut, skin-educated cells return to the skin, and systemically active cells patrol the secondary lymphoid organs. The multi-step adhesion cascade that governs lymphocyte homing, first characterized for rolling on the high endothelial venules of lymph nodes and extended to all endothelial beds, involves 4 sequential, partially independent steps, tethering and rolling, mediated by selectins interacting with glycoprotein ligands, activation, mediated by chemokine signaling from the endothelial surface, stable adhesion, mediated by integrin-ICAM interactions, and transmigration through the endothelial monolayer. Each step must be completed before the next can proceed, creating a series of checkpoints that ensure that only the right cells transmigrate into any given tissue. The tissue specificity of this cascade arises from the expression of different addressins by different endothelial beds. Peripheral lymph node HEVs express PNAD, recognized by L-selectin. Gut lamina propria endothelium expresses MADCAM1, recognized by alpha-4 beta-7 integrin. Skin dermal endothelium expresses E-selectin and VCAM1, recognized by cutaneous lymphocyte antigen and alpha-4 beta-1 integrin. Lymphocytes that have been activated in the gut by the retinoic acid-rich environment of the mesenteric lymph nodes upregulate alpha-4 beta-7 and CCR9, the receptor for the gut chemokine CCL25, ensuring that they home preferentially to the gut on their subsequent trafficking. Lymphocytes activated in the skin-draining lymph nodes are imprinted by a different signal through the production of vitamin D metabolites and cutaneous chemokines to express the homing receptors that direct them back to the skin. This tissue-specific imprinting of lymphocyte homing is the basis of the concept of the mucosal immune system as a partly separate compartment from the systemic immune system. The splenic marginal zone, the region between the white pulp and the red pulp, home to marginal zone B cells, marginal zone macrophages, and NK cells, is the site where the blood's contents are first sampled by the immune system, and its specialized cellular composition is adapted for the rapid, T-independent responses to blood-borne pathogens that are the spleen's most distinctive immunological contribution. The marginal zone is the spleen's immunological front line, the region where the blood, arriving through the central arterioles, first encounters the immune cells that will assess its contents and respond if a threat is detected. The blood flow in the marginal zone is characterized by a slowing from the rapid arterial flow of the central arteries. As the vessels open into the marginal sinuses, that creates the conditions for the efficient capture of blood-borne antigens and pathogens by the marginal zone macrophages. The marginal zone macrophages express a distinct pattern of scavenger receptors and pattern recognition receptors, including SINR1, which captures polysaccharide antigens, and the mannose receptor, that equip them to capture and process the types of antigens commonly carried in the blood during bacteremia. These macrophages can relay captured antigen to the adjacent marginal zone B cells, activating the T-independent B cell responses that produce rapid IgM against blood-borne pathogens. The NK cells of the marginal zone are also strategically positioned to kill any cells that have been identified as aberrant, missing self by the NK cell's inhibitory receptor system, from the blood flowing through. The structural integrity of the marginal zone, its physical organization and the density of its cellular populations, is a determinant of the spleen's effectiveness as a blood filter, and is disrupted in the hyposplenism that can accompany conditions including sickle cell disease, where repeated infarctions damage the splenic parenchyma, celiac disease, where the marginal zone is specifically depleted, and essential thrombocythemia, where megakaryocyte infiltration disrupts the normal architecture. The gut's lymphatic system is modified from the systemic lymphatic system in having lacteals rather than conventional lymphatic capillaries in the villus, reflecting the unique demands of fat absorption, and the structure and function of the lacteals is regulated by the same VEGF-C-VEGFR3 pathway that governs systemic lymphangiogenesis. The intestinal lacteal, the central lymphatic capillary of each intestinal villus, is adapted for its specialized function of chylomicron absorption in ways that distinguish it from systemic lymphatic capillaries. The lacteal is a single, blind-ended tube that runs along the central axis of the villus from tip to base, connected at its base to the submucosal collecting lymphatics that drain toward the mesenteric nodes. The lacteal's endothelial cells form button junctions, as described for lymphatic capillaries generally, but the lacteals also have a uniquely open structure at their tips, where chylomicron particles can enter through the loosely organized endothelial cell-cell junctions, and a rich coverage by the smooth muscle extensions of the villus, which compress the lacteal rhythmically as the villus contracts during food transit, propelling the chylomicron-laden chyle toward the collecting lymphatics. The lacteals are maintained by the VEGF-C produced by smooth muscle cells and stromal cells of the villus, acting on VEGFR3 on the lacteal endothelium, the same signaling axis that drives systemic lymphangiogenesis. Mice lacking VEGFR3 signaling in the intestinal lymphatics develop chylous ascites and steatorrhea, confirming the essential role of this pathway in lacteal maintenance. The lacteals also respond to dietary fat. A high-fat meal increases the VEGF-C signaling in the intestinal villi and promotes the opening of the lacteal junctions and the expansion of the lacteal lumen, transiently increasing the lacteal's capacity to accommodate the increased chylomicron flux of the post-meal period. The lymphatic vessels of the heart, the cardiac lymphatics, drain the myocardial interstitial fluid and are essential for maintaining the cardiac interstitial environment, and their obstruction or dysfunction, as can occur after cardiac transplantation, after myocardial infarction, or in the context of cardiac inflammation, contributes to myocardial edema and impaired cardiac function. The heart is a metabolically extraordinary organ, producing enormous amounts of metabolic waste during its continuous mechanical work. In the cardiac lymphatics, the network of lymphatic capillaries, precollectors, and collectors that drain the myocardial interstitium, are essential for clearing this waste and maintaining the interstitial fluid balance that allows the cardiomyocytes to function normally. The cardiac lymphatics originate as blind-ended lymphatic capillaries in the subepicardial connective tissue and in the myocardium itself, drain to the precollectors in the epicardial tissue, and converge on the main cardiac lymphatic trunks that ascend alongside the coronary arteries and ultimately drain into the mediastinal lymph nodes and the thoracic duct. The propulsion of cardiac lymph is assisted by the mechanical pumping action of the heartbeat itself. The rhythmic compression and relaxation of the myocardium provides the intrinsic pumping force for the cardiac lymphatics in a way that skeletal muscle provides it for the limb lymphatics. After cardiac transplantation, the cardiac lymphatics are disrupted at the sites of surgical anastomosis, and the cardiac allograft is initially without lymphatic drainage. The subsequent re-establishment of cardiac lymphatic drainage, through the invasion of lymphatic sprouts from the recipient's mediastinal lymphatics, takes weeks to months, during which the transplanted heart is at increased risk of myocardial edema and potentially of impaired immune surveillance of the graft, contributing to the immunological challenges of the immediate post-transplantation period. The cytotoxic mechanisms of CD8 T cells. The granule exocytosis pathway delivering perforin and granzymes to the target cell, and the phosphoryl pathway triggering apoptosis, are deployed at the immune synapse between the cytotoxic T cell and its target, ensuring that the cytotoxic machinery is directed precisely at the identified target cell without damaging surrounding cells. The killing of infected or tumor cells by CD8 cytotoxic T cells, one of the most powerful effector mechanisms of the adaptive immune system, is achieved through a remarkably targeted process that confines the cytotoxic machinery to the interface between the effector cell and its target, preventing the collateral damage to surrounding normal cells that would otherwise accompany the release of potent cytotoxic molecules. The cytotoxic granules of the CD8 T cell, membrane-bound vesicles containing perforin and granzymes, are initially distributed throughout the cell's cytoplasm. Upon specific recognition of its target through the T-cell receptor, the CD8 T cell forms an immune synapse with the target cell, and the microtubule organizing center of the T cell reorients to face the synapse, a polarization that directs the cytotoxic granules to the synapse interface. The granules fuse with the T cell membrane specifically at the synapse, releasing perforin and granzymes into the closed space of the synaptic cleft between the two cells. Perforin, in the presence of calcium, polymerizes to form pores in the target cell membrane, large enough for the granzyme proteins to enter. Granzyme B, the most important granzyme, enters the target cell through the perforin pores and activates the caspase cascade, triggering the apoptosis, the programmed self-destruction, of the target cell. The entire lethal process, from granule exocytosis to target cell death, is completed in approximately 5 minutes. The T cell then disengages, its granules having been spent on this target, and can rearm and kill another target cell. The phenomenon of immune evasion by HIV, the virus that causes AIDS, specifically targets the CD4 T helper cell, the central coordinator of the adaptive immune response, and its progressive destruction of this population produces the combined failure of cellular and humoral immunity that is the defining feature of the acquired immunodeficiency syndrome. The strategic genius of HIV's immune evasion, if such anthropomorphic framing is permissible, lies in the identity of its primary target, the CD4 T helper cell, the cell whose cooperation is required for virtually every arm of the adaptive immune response. HIV enters CD4 T cells by binding simultaneously to the CD4 molecule and a coreceptor, CCR5 or CXCR4, on the cell's surface, fusing with the cell membrane and injecting its RNA genome and enzymes into the cytoplasm. The reverse transcriptase enzyme converts the viral RNA to double-stranded DNA, which integrates into the host cell's genome as the HIV provirus. From this integrated position, the virus can remain latent for years in resting memory T cells, escaping immune detection because the latent provirus produces no viral proteins, or it can replicate actively, producing new virions that bud from the infected T cell and go on to infect further CD4 T cells. The progressive depletion of CD4 T cells, through direct viral killing, through cytotoxic T cell killing of infected CD4 cells, and through the bystander death of uninfected CD4 T cells in the chronically inflamed lymph nodes of untreated HIV infection, produces the multi-system immune deficiency of AIDS. Without CD4 T helper cells, cytotoxic T cells cannot receive the help they need for full activation and memory. B cells cannot receive the germinal center help they need for affinity maturation and class switching. Macrophages cannot be activated to kill intracellular pathogens. The result is susceptibility to the opportunistic infections, pneumocystis pneumonia, cryptococcal meningitis, CMV retinitis, Kaposi's sarcoma, that define the clinical syndrome of AIDS. The lymph node's architecture is disrupted in diseases that infiltrate the node, from lymphoma, from metastatic carcinoma, from granulomatous infections including tuberculosis and sarcoidosis, and the clinical detection of lymphadenopathy requires the correlation of node size, texture, distribution, and associated symptoms with the wide differential diagnosis that lymph node enlargement represents. The lymph node is, as an immune organ, designed to respond to antigenic stimulation by enlarging. The follicular hyperplasia, the germinal center formation, and the paracortical expansion of a reactive lymph node represent the node doing exactly what it was designed to do. But lymph node enlargement also occurs in conditions that the node was not designed to handle: the proliferation of malignant lymphoma cells within the node, which replace the normal lymphoid architecture with sheets of monoclonal malignant lymphocytes, the deposition of metastatic carcinoma cells in the sinuses and parenchyma of the node, which arrive from the tumor's primary site via the afferent lymphatics, and the granulomatous inflammation of tuberculosis and sarcoidosis, which produces the characteristic epithelioid granulomata with central caseating necrosis in the former and non-caseating granulomata in the latter. The clinical evaluation of lymphadenopathy, lymph node enlargement, requires the integration of multiple clinical parameters: the size, nodes over 1 centimeter in diameter merit investigation; the texture, firm and rubbery in lymphoma, hard and fixed in metastatic carcinoma, tender and reactive lymphadenitis; the distribution, regional versus generalized, and the associated symptoms, fever, night sweats, and weight loss; the B symptoms of lymphoma; the duration and pace of enlargement, the age of the patient, and the associated infectious and systemic symptoms all further refine the differential diagnosis. The enlarged lymph node that persists without an obvious infectious cause, particularly in a patient with systemic symptoms or in the older adult, warrants histological assessment. The phenomenon of the cytokine storm, the dysregulated, excessive immune response in which the feedback loops that normally limit cytokine production fail, producing a cascade of escalating inflammation that damages the organs it was designed to protect, is the mechanism of the most severe complications of influenza, COVID-19, and CAR T-cell therapy. The cytokine storm is the immune system's most dangerous failure mode, a state in which the normal negative feedback mechanisms that terminate the inflammatory response have been overcome, and the cytokine-producing cells of the immune system enter a positive feedback loop of mutual activation that drives the inflammatory signal progressively higher rather than resolving it. In severe influenza pneumonia, a highly pathogenic influenza virus in the lower respiratory tract triggers a massive innate immune response in the alveolar macrophages and epithelial cells, the production of type I interferons and pro-inflammatory cytokines at concentrations far exceeding the normal response to upper respiratory infection. These cytokines attract enormous numbers of monocytes and neutrophils from the circulation, which are themselves activated to produce more cytokines, further amplifying the response. The resulting inflammatory damage to the alveolar epithelium, the very tissue that the immune response is trying to protect, impairs gas exchange and produces the acute respiratory distress syndrome. In severe COVID-19, a similar amplification occurs, with additional contributions from the complement system and the coagulation cascade. In CAR T-cell therapy, in which patient T-cells are engineered to carry chimeric antigen receptors that recognize tumor antigens, the dramatic activation of the engineered T-cells upon tumor contact triggers the rapid release of enormous quantities of cytokines, producing the cytokine release syndrome that can be life-threatening if not managed with the interleukin-6 receptor blocker tocilizumab. The cytokine storm reveals both the power of the immune system and the cost of that power when its regulatory mechanisms fail. The secondary lymphoid organs, the lymph nodes, the spleen, the mucosa-associated lymphoid tissue, and the tertiary lymphoid structures that develop in chronically inflamed tissues, all share the common function of concentrating antigen-specific lymphocytes in a physical microenvironment that maximizes the probability of the antigen receptor encounter that initiates the adaptive immune response. The secondary lymphoid organs are, in their underlying function, all solutions to the same problem: how to bring together the rare lymphocyte specific for a particular antigen with the antigen itself and with the antigen-presenting cells that activate it, in a physical space and at a concentration that makes the encounter probable rather than vanishingly unlikely. The diversity of secondary lymphoid organ forms, the anatomically fixed lymph nodes of the subcutaneous and perivisceral tissues, the unique blood-filtering architecture of the spleen, the follicular aggregates of the Peyer's patches and tonsils, the diffuse lymphoid tissue of the lamina propria, reflects the diversity of the routes by which antigens enter the body and the diversity of the environments in which immune surveillance is required. Tertiary lymphoid structures, organized lymphoid tissue that forms de novo in sites of chronic inflammation, including the chronically inflamed joints of rheumatoid arthritis, the inflamed plaques of autoimmune thyroiditis, and the tumor microenvironment of some cancers, represent the immune system extending the secondary lymphoid organ function into the tissues where the inflammatory stimulus is present, bringing the germinal center machinery directly to the site of the pathology. The formation of tertiary lymphoid structures in the tumor microenvironment, detectable by the presence of dense T-cell and B-cell infiltrates with evidence of germinal center activity, is associated with improved prognosis and better response to checkpoint inhibitor immunotherapy in several cancers, suggesting that these structures are sites of anti-tumor immune responses that may be therapeutically significant. The lymphatic system's role in maintaining tissue fluid homeostasis is dynamically regulated in response to physiological challenges, increasing lymph flow in response to exercise, inflammation, and increased capillary filtration, and reducing flow during rest, through mechanisms that include the stretch-sensitive contraction of lymphangin smooth muscle and the modulation of lymphangin tone by autonomic signals. The lymphatic system's ability to match its drainage capacity to the fluid load presented by the tissues is essential for the prevention of edema under the widely varying conditions of physiological life. During vigorous exercise, the increased cardiac output dramatically increases capillary filtration. More blood is being pumped through the capillary beds at higher pressure, driving more fluid into the interstitium. Simultaneously, the working muscle's rhythmic contractions provide an enhanced mechanical pump for the deep lymphatics of the limb, increasing lymphatic flow. The inflammatory response to tissue injury similarly increases capillary filtration through the prostaglandin and histamine-mediated increase in capillary permeability, and the resulting increase in interstitial fluids stimulates lymphatic contraction through the stretch-sensitive response, increasing lymph flow and drainage. Under resting conditions, as at night, as during sleep, the reduced cardiac output and reduced capillary filtration reduce the fluid load on the lymphatics, and the autonomic nervous system's parasympathetic dominance reduces lymphatic contraction frequency, allowing the lymphatic system to operate at a lower, more energy-efficient pace. The match between tissue fluid production and lymphatic drainage capacity that this dynamic regulation maintains is what keeps the tissues free of edema under the enormous range of physiological conditions that daily life imposes. Right now, as you lie here in the quiet, the lymphatic system is operating at its most relaxed, the fluid load minimal, the lymphatic contraction slow, the drainage proceeding at its gentlest pace. The body at rest, the lymph flowing quietly. Everything as it should be. You have stayed with me a very long time tonight. Through the capillaries and the nodes and the germinal centers and the dendritic cells and the B cells and the T cells and the complement cascade and the natural killers and the mucosal immunity and all the quiet, tireless biology of the lymphatic system. 100 facts, and you have been here for nearly all of them. I do not take that lightly. Time is the most honest gift there is. If Relax with Science has earned a small place in your evenings, if these nights have given you something you would like to return to, subscribing is the simplest and most honest way to say so. I will be here. I always will be. Just a little further. The night is nearly here completely. You have stayed with me a very long time tonight. Through the capillaries and the nodes and the germinal centers and the dendritic cells and the B cells and the T cells and the complement cascade and the natural killers and the mucosal immunity and all the quiet, tireless biology of the lymphatic system. 100 facts, and you have been here for nearly all of them. I do not take that lightly. Time is the most honest gift there is. If Relax with Science has earned a small place in your evenings, if these nights have given you something you would like to return to, subscribing is the simplest and most honest way to say so. I will be here. I always will be. Just a little further. The night is nearly here completely. The NK cell's ability to kill without prior sensitization, its ready-state killing of cells that have lost MHC class I expression, makes it the immune system's rapid response tumor surveillance mechanism, and the NK cell infiltration of solid tumors is associated with improved prognosis in many cancer types, reflecting the ongoing anti-tumor NK cell activity in the tumor microenvironment. Natural killer cells are among the most important anti-tumor immune cells precisely because they do not require the prior antigen exposure and clonal expansion that take days to weeks for cytotoxic T cells. An NK cell that encounters a tumor cell expressing stress ligands and lacking normal MHC class I can kill that tumor cell immediately, without any preceding sensitization. This killing-ready state makes NK cells the lymphatic immune system's first-line anti-tumor surveillance mechanism, capable of responding to early-stage transformed cells before the adaptive immune system has had time to develop antigen-specific responses. The clinical evidence for NK cell anti-tumor activity is substantial. Patients with higher NK cell numbers in the peripheral blood and in tumor infiltrates have better outcomes in many cancers including colorectal cancer, lung cancer, renal cell carcinoma, and gastric cancer. The tumor microenvironment, however, often impairs NK cell function through the same immunosuppressive mechanisms that impair T cell function: regulatory T cells, M2 macrophages, and the TGF-beta and PD-L1 expression of tumor cells. NK cell dysfunction in the tumor microenvironment can be reversed by cytokines: interleukin 15, interleukin 12, interleukin 2, that restore NK cell cytotoxicity, and the therapeutic administration of these cytokines or of engineered NK cells is being investigated as a complement to the checkpoint inhibitor immunotherapy that primarily targets T cells. The NK cell's unconditional readiness to kill makes it a compelling target for cancer immunotherapy. The immune system's ability to respond to influenza vaccination varies markedly between individuals and across the lifespan, with factors including age, prior influenza exposure history, vaccine formulation, and genetic variation in immune response genes all contributing to the variable vaccine-induced antibody titers and clinical protection observed in population studies. Influenza vaccination is one of the most extensively studied and most difficult vaccination programs in medicine. Not because the science of influenza immunity is unknown, but because the variability of the immune response and the variability of the viral antigen make it a perpetually imperfect match between the immune protection generated and the protection actually needed. In young, immunologically naive individuals receiving their first influenza vaccination, the immune response is often vigorous. The germinal center reaction generates high-affinity antibodies through affinity maturation, and the seroconversion rate is high. In the elderly, the same vaccine produces lower antibody titers, lower avidity antibodies, and reduced cellular immune responses, reflecting the immunosenescence of the aged immune system, the smaller germinal centers, the reduced B cell diversity, the impaired T helper cell function. The prior influenza exposure history of the individual, the original antigenic sin phenomenon, shapes which epitopes the antibody response targets, potentially misdirecting it toward conserved epitopes of old strains rather than the novel epitopes of the current strain. Genetic variation in the HLA genes, which determine which peptides can be presented to T cells, and in the pattern recognition receptor genes and signaling pathway genes contributes to the individual variability in vaccine immunogenicity that is observed even in young, healthy adults. Understanding this variability is essential for the development of the universal influenza vaccine, A vaccine targeting conserved influenza antigens that would not require reformulation each year and would provide protection against pandemic strains. That remains one of the major goals of influenza immunology research. The lymphatic system of the skin, the cutaneous lymphatics, plays an important role in the pathogenesis of psoriasis, where the lymphatic vessels are dilated and dysfunctional in psoriatic plaques, contributing to the impaired drainage and chronic inflammatory milieu of the disease, and where therapeutic restoration of lymphatic function may represent a novel treatment approach. Psoriasis, the immune-mediated inflammatory skin disease characterized by well-demarcated erythematous, scaling plaques, has been recognized for some time as a disease of immune dysregulation, with the pathological activation of Th17 cells and their production of interleukin-17 as the central immunological driver. Less appreciated has been the role of the cutaneous lymphatic system in the disease's pathogenesis. The lymphatic vessels in psoriatic plaques are abnormal. They are dilated beyond their normal caliber, have impaired contractile function, and show evidence of reduced valve integrity that allows backflow rather than the unidirectional flow of healthy lymphatics. These functional impairments reduce the ability of the psoriatic skin's lymphatics to drain the edema, inflammatory mediators, and immune cells that the intense inflammatory response produces, creating a stagnant inflammatory environment that perpetuates the immune activation. Research in psoriasis models has shown that pharmacological stimulation of lymphangiogenesis through VEGF-C, or restoration of lymphatic contractility, can reduce the inflammatory burden in the skin and alleviate the disease severity, suggesting that targeting the lymphatic dysfunction may provide therapeutic benefit independent of or complementary to the current immune targeting approaches. The psoriatic plaque is a failure of lymphatic resolution, rather than merely a failure of immune regulation, represents a shift in perspective that may open new therapeutic avenues. The concept of trained innate immunity, the ability of innate immune cells including macrophages and NK cells to develop a heightened, more effective state of response following prior antigen exposure, through epigenetic reprogramming rather than clonal selection, challenges the traditional view that immunological memory is exclusively a property of the adaptive immune system. The distinction between innate and adaptive immunity has traditionally been drawn on the axis of memory. Innate immunity responds immediately but without learning, while adaptive immunity learns from experience and remembers. Trained innate immunity, described primarily through the work of Mihai Neghia and colleagues, challenges this clean distinction by demonstrating that monocytes, macrophages, and NK cells can develop an enhanced, more effective response to pathogens following prior exposure, not through clonal selection and receptor rearrangement, but through epigenetic reprogramming of the cell's gene expression. Monocytes that have been trained by exposure to beta-glucan, a fungal cell wall component, or by BCG vaccination, the tuberculosis vaccine, show increased production of pro-inflammatory cytokines in response to unrelated stimuli such as LPS, and enhanced killing of bacteria and fungi for weeks to months after the training stimulus. This enhanced responsiveness is not antigen-specific. A hallmark of trained innate immunity is that training with one stimulus can enhance responses to unrelated stimuli, and it is mediated by epigenetic changes in the chromatin of the trained cells, increased histone acetylation and methylation at the promoters of cytokine genes, creating an open chromatin configuration that allows more rapid and more robust transcriptional activation when the genes are subsequently stimulated. Trained innate immunity may explain the nonspecific benefits of certain vaccines, including the reduction in non-tuberculosis mortality observed in BCG-vaccinated infants, and it provides a new conceptual framework for understanding how prior infection and vaccination can enhance immunity to subsequent unrelated pathogens. The mesenteric lymph nodes, the largest lymph node cluster in the body, containing approximately 100 to 150 nodes in the adult human, are the site at which the immune response to the gut's enormous antigen load is primarily conducted, and their unique immunological environment, shaped by the gut microbiome and by retinoic acid from the intestinal epithelium, produces the regulatory, tolerogenic immune responses appropriate for the gut's microbially rich environment. The mesenteric lymph nodes are, in terms of sheer immunological workload, the hardest working lymph nodes in the body, continuously processing the antigens delivered by the mesenteric lymphatics from the gut's teeming microbial content, the dietary antigens of every meal, and the cellular debris and metabolic products of the most active and rapidly renewing epithelial surface in the body. The immunological character of the mesenteric node is shaped from above by the intestinal environment, the dendritic cells that arrive from the gut, conditioned by the retinoic acid produced by the intestinal epithelium and by the microbial signals of the mucosal surface, carry with them not only their antigenic cargo but the molecular imprinting that will direct the node's immune response. Retinoic acid instructs the dendritic cells to promote the differentiation of regulatory T cells, expressing FOXP3 and producing TGF-beta and IL-10, rather than the Th1 and Th17 effector cells that would drive intestinal inflammation. Retinoic acid also imprints the gut homing phenotype on the T cells that are activated in the mesenteric nodes, upregulating alpha-4 beta-7 integrin and CCR9, so that the tolerogenic T cells generated in the mesenteric nodes are directed back to the gut, where they continue their regulatory function. This local immunological architecture, the retinoic acid-rich environment, the regulatory T-cell bias, the gut homing imprinting, is the molecular basis of the gut's remarkable capacity for immune tolerance, and its disruption, by dysbiosis, by vitamin A deficiency, by inflammatory conditions, is a mechanism through which intestinal inflammation and inflammatory bowel disease can develop. The lymphatic system's role in the establishment and maintenance of the normal gut microbiome, through the IgA that the lamina propria plasma cells produce and transport across the epithelium into the gut lumen, provides a mechanism by which the adaptive immune system shapes the microbial community of the gut through selective immunological pressure. The secretory IgA that lines the mucosal surfaces of the gut, produced at a rate of approximately 3 to 5 grams per day, making it by mass the most abundantly produced antibody in the body, is not simply a defense against pathogens but a selective immunological force that shapes the composition and organization of the gut microbiome. The IgA produced in the lamina propria plasma cells is transported across the epithelium by the polymeric immunoglobulin receptor, which binds the dimeric IgA, transcytoses it, and cleaves to release the secretory component attached IgA into the gut lumen. This secretory IgA then coats bacterial cells, both pathogens and commensals, in a process called immune exclusion that prevents their direct adherence to the epithelial surface and reduces their access to the epithelial border region where they could stimulate immune responses. IgA coating does not necessarily kill the coated bacteria. It is non-inflammatory by virtue of lacking complement activating function, but it alters their spatial distribution in the gut, confining them to the lumen rather than the mucus layer closest to the epithelium. Different bacterial species in the gut microbiome are coated by IgA to different degrees, and the IgA specificity appears to be adaptive, targeting the most immunogenic or potentially pathogenic members of the microbiome more heavily than the most commensal and beneficial ones. This selective IgA coating shapes the competitive landscape of the gut microbiome, providing immunological support to the bacteria that are best coated, which are often the most mutualistic and least inflammatory members of the community. The lymphatic system's involvement in aging. Through the progressive deterioration of lymph node architecture, the accumulation of adipocytes in the lymph node parenchyma, adipose tissue replacement, the reduced germinal center activity, and the impaired lymphatic vessel contractility of the elderly contributes to the immunosenescence and reduced fluid balance regulation that characterize the aging body. The lymphatic system, like every other organ system, is subject to the structural and functional changes of normal aging. And because the lymphatic system is both an immune organ and a fluid regulatory system, the consequences of these changes are felt in both domains. The lymph node in old age shows several characteristic changes: the replacement of the normal lymphoid follicle and paracortex by adipose tissue, the same process of adipose involution that affects the thymus, extended to the peripheral lymph nodes, reduces the density of lymphocytes in the supporting stroma in the node and reduces the node's capacity for germinal center formation. The remaining germinal centers in aged nodes are smaller, produce less affinity-matured antibody, and generate fewer long-lived plasma cells and memory B cells, contributing to the reduced vaccine immunogenicity of old age. The lymphatic vessels themselves show reduced contractile capacity with aging. The lymphangian smooth muscle becomes less responsive to the stretch stimulation that drives contraction frequency, and the intrinsic pacemaking frequency falls. This reduced lymphangian function impairs lymphatic drainage and may contribute to the increased prevalence of tissue edema in the elderly, particularly in the lower extremities where the gravitational challenge to lymphatic return is greatest. The combination of impaired immune function and impaired fluid regulation, both downstream of the aging lymphatic system, contributes to the frailty, reduced infection resistance, and altered body fluid distribution that are characteristic features of very old age. The phenomenon of B-cell editing. the process by which immature B cells in the bone marrow that express self-reactive antibody receptors can modify their receptor specificities by secondary VJ recombination rather than simply dying by negative selection, provides an additional mechanism for purging the self-reactive repertoire while preserving the B cell's potential. Negative selection. The deletion of self-reactive lymphocytes in the primary lymphoid organs is the primary mechanism of central tolerance, but the discovery of receptor editing has revealed an alternative fate for some self-reactive B cells that provides the immune system with a way of rescuing potentially self-reactive cells by changing their receptors rather than simply eliminating them. When an immature B cell in the bone marrow has its B cell receptor cross-linked by a self-antigen, the signal that triggers negative selection, it has two possible fates. If the cross-linking is sustained and strong, the cell initiates apoptosis, negative selection. But if the cross-linking is mild or transient, the cell can instead reactivate its RAG recombinase, the enzyme responsible for VJ recombination during B cell receptor gene assembly, and rearrange a different combination of immunoglobulin light chain gene segments. If this secondary rearrangement produces a new light chain that, combined with the existing heavy chain, creates a receptor that no longer recognizes the self-antigen, the B cell has edited itself into a non-self-reactive cell and can continue its development. If the secondary rearrangement continues to produce self-reactive receptors, the cell eventually undergoes apoptosis. Receptor editing is estimated to account for approximately 25% of the self-reactive B cells in the bone marrow being rescued by changing their specificity rather than being deleted. It provides the immune system with a mechanism to preserve B cells with potentially useful heavy chains, whose variable regions may be valuable in the repertoire, even if the first light chain combination produces self-reactivity. The lymphatic system's contribution to the body's homeostasis is perhaps most visible in its absence, in the lymphedema that follows disruption, in the immunodeficiency that follows lymphocyte depletion, in the fat malabsorption that follows lacteal failure. But its ordinary, uninterrupted function is the invisible foundation on which the body's fluid balance, immune defense, and nutritional delivery are built every day of life. The lymphatic system is, by its nature, a system of negatives. It is noticed in its absence, appreciated in its failure, studied in its disruption. In health, it is perfectly invisible. The fluid it drains leaves no trace of its passage. The immune cells it transports arrive at their destinations by routes that no sensation reports. The fat it absorbs from the gut reaches the bloodstream without any awareness on the part of the person being nourished. The lymph nodes that filter the lymph for threats do their work below any threshold of sensation unless they enlarge enough to be felt as lumps. And even then, the work they are doing, the germinal center reactions, the T cell activations, the antibody calibrations, is known only to those who examine the node's tissue under a microscope. This invisibility is not a limitation of the lymphatic system. It is one of its primary features. A system that does its work without drawing attention to itself is a system that allows the conscious mind to devote itself to other things, to the day's work, to the world's beauty, to the conversation at hand, while the body manages its own maintenance. The lymphatic system's silence is the silence of competence. The silence of a system that has been working correctly for so long that it no longer needs to announce itself. And tonight, as you lie here and the day dissolves and the body settles into its own quiet restoration, the lymphatic system continues its work, draining the fluid, filtering the lymph, running its immune surveillance, returning the fat of the day's last meal to the blood, in the perfect silence of something that has never needed your attention because it has always had its own. The lymphatic system is the body's most patient and most persistent hidden infrastructure, neither as dramatic as the heartbeat nor as immediate as the neural signal, but as necessary as either, and as continuous, and as extraordinary, once you have been given the language to appreciate what it quietly does. Begin with the simplest version of this last fact, right now, in the tissues of your body, in the skin, in the organs, in the fascia between the muscles and beneath the skin. The lymphatic capillaries are collecting the fluid that leaked from the capillaries of the cardiovascular system, collecting the cells of the immune system that have been patrolling the tissue and are now returning, collecting the cellular debris of the day's normal cell turnover, the metabolic waste, the proteins too large for the blood capillaries to reabsorb. And somewhere in the intestine, in the lacteals of the villi, collecting the last of the day's dietary fat, the chylomicrons still in transit from the last meal, making their way toward the cisterna chyli and the thoracic duct and ultimately the venous blood. The lymph nodes, 600 to 800 of them, scattered through the body like small, quiet stations on a surveillance network, have been sampling the day's antigenic traffic, checking it against the accumulated immunological memory of a lifetime, noting anything that requires attention. The germinal centers that may be active in some of those nodes have been running their extraordinary process, the somatic hypermutation, the selection, the affinity maturation, refining the antibody response to whatever the body has encountered. The regulatory T cells have been exerting their quiet governance, preventing the immune system from turning against the body's own tissues. The natural killer cells have been patrolling for the cellular signatures of infection and transformation. All of this, all of it, right now, in the same body that is lying here and breathing slowly and moving towards sleep. The lymphatic system is not something the body has in addition to everything else that keeps it alive. It is part of everything else. It is the drainage and the defense and the delivery, the quiet river running through the entire body, carrying what is needed, clearing what is not, keeping the balance that life requires. Let it do its work. It has been doing it your whole life. It will do it tonight, and tomorrow, and for every day that the body continues. The river runs. The balance holds. The night is yours. The lymphatic system you have been traveling through tonight is not separate from you. It is not a subject held at a careful academic distance. Every vessel we described is running through your body right now. Every lymph node we visited is doing its quiet work in your neck, in your chest, in your abdomen, in your groin. The lymphocytes we talked about are circulating in your blood and your lymph, recirculating through the nodes, patrolling the tissues. The germinal centers, if any are active in your nodes right now, are doing their extraordinary molecular work, mutating and selecting and refining the antibody that will protect you from something you have recently encountered and will not forget. The lacteal system, in the quiet intestine, is still moving the day's last fat toward the thoracic duct. All of this, in the same body, in the same darkness, at this same hour. The lymphatic system does not stop when you do. It is more nocturnal than most of the body. The lymphatic clearance that runs best during sleep, the immune surveillance that continues through the night, the lymphangian contractions that move the day's accumulated fluid back toward the heart while the muscular pump has gone quiet. The lymphatic system, in some ways, belongs to the night. Somewhere in the course of these 100 facts, the body has settled. The breath has found its slower rhythm. The heart is doing the quiet work of rest. The lymph is moving at its most gentle pace, the fluid load minimal, the drainage proceeding without effort. The immune system is running its night shift, the natural killer cells and the macrophages maintaining their surveillance at a lower metabolic cost than the daytime fever of the active inflammatory response. The germinal centers, if they are active, are working in the dark interior of the lymph nodes in the same indifference to the hour that they always maintain. The body's repair work is underway. The cells that divide most rapidly at night, the skin and the gut epithelium and the bone marrow producing their replacements, and the lymphatic system is clearing the path for this renewal, draining what needs to be cleared, delivering what needs to be delivered. This is Relax with Science, saying goodnight, and meaning it with the full weight of everything we have moved through together. The wonder that was in these facts does not leave when the words stop. It remains in the quiet river of the lymphatic system, moving through the body as it has always moved. The body you heard about tonight is the body you are lying in. The biology we described is the biology that is sustaining you. And the lymphatic system, that patient, invisible, indispensable infrastructure, is doing what it has always done, without announcement and without credit, in the service of keeping you here, breathing, whole. You are the fluid being returned. You are the antigen being surveyed. You are the balance being maintained. You are, in the most literal possible sense, the lymphatic system at rest. Let the river run. It has always known the way.